Diri atas para peneliti dan prekayasa terlatih, tim logistik, dan tim administrasi. Diri atas para peneliti dan prekayasa. Melalui fasilitas laboratorium BSL-2 dan BSL-3 yang dimiliki, LIPI siap berkontribusi secara maksimal untuk mendukung pemerintah dalam mempercepat penanganan COVID-19. Sahabat LIPI dimanapun berada, jangan lengah, jangan menyerah, dan jangan lupa untuk selalu mematuhi protokol kesehatan dimanapun berada ya. Bersama, kita bisa melawan pandemi ini. Salam sehat dan semangat ya! Selamat datang di video tour Biosafety Level 2 dan Biosafety Level 3 LIPI. Lembaga Ilmu Pengetahuan Indonesia atau LIPI merupakan lembaga penelitian yang memiliki fasilitas laboratorium Biosafety Level 2 atau BSL-2 dan Biosafety Level 3 atau BSL-3 yang terletak di kawasan Cibinong Science Center, Bogor. Laboratorium ini digunakan untuk melaksanakan kegiatan penelitian yang melibatkan organisme dan patogen yang berbahaya. Saat ini, kedua laboratorium tersebut telah ditetapkan sebagai laboratorium pemeriksa COVID-19. Biosafety Level, atau biasa disingkat BSL, adalah suatu tingkat sistem keamanan yang dibuat untuk menangani bahan biologi berbahaya di laboratorium. Tujuannya untuk melindungi personil yang bekerja di dalam laboratorium, juga lingkungan dan komunitas sekitar dari paparan mikroba patogen yang sedang diteliti atau diuji. BSL memiliki 4 tingkatan, dari 1 sampai 4, di mana semakin tinggi tingkatannya, maka semakin khusus fasilitasnya, dan semakin berbahaya bahan biologi yang dapat ditangani di fasilitas tersebut. Perbedaan utama dari tingkatan ini adalah sistem tata udara, tekanan, kelengkapan fasilitas, dan alat penunjang lainnya. Kita ke laboratorium BSL-2 dulu yuk. Di dalam BSL-2 ini, ada banyak ruangan yang digunakan sesuai fungsinya untuk deteksi virus SARS-CoV-2 sebagai penyebab pandemi COVID-19. Ruangannya terdiri dari 1. Ruang penerimaan spesimen. 2. Ruang logistik sebagai tempat penyimpanan bahan kimia dan bahan habis pakai. 3. Ruang ganti untuk pemakaian alat pelindung diri. 4. Lab ekstraksi yang terdiri atas ruang antara, ruang laboratorium, ruang mandi, dan ruang dekontaminasi limbah. 5. Lab PCR yang terdiri atas ruang antara, ruang pre-PCR, ruang post-PCR, dan ruang analisis. Nah, kalau tadi sahabat LIPI sudah ke BSL-2, yuk sekarang kita ke BSL-3. Fasilitas laboratorium BSL-3 LIPI memiliki infrastruktur yang lengkap dan telah tersertifikasi sesuai standar WHO sejak tahun 2018 dan terus diperbaharui setiap tahun. Biosafety Level 3 memiliki 4 laboratorium, yaitu Clean Rooms, yang terdiri dari Health Biotechnology Laboratory, Food Biotechnology Laboratory, Biosafety Lab, terdiri dari BSL-3 Laboratory, Animal BSL-3 Laboratory. BSL-3 LIPI terdiri atas 2 ruangan. 1. Ruangan mikroba. Ruangan ini digunakan memperbanyak atau mengisolasi materi genetik bakteri untuk digunakan dalam kegiatan penelitian. Misalnya, pencarian obat baru, vaksin, dan sebagainya. 2. Ruangan kultur sel. Ruangan ini digunakan untuk mengkultivasi virus di dalam sel mamalia hidup. Sedangkan Animal BSL-3 terdiri atas 2 ruangan. 1. Ruangan isocages. Ruangan ini digunakan untuk memelihara hewan pengerat yang digunakan sebagai model pada uji di laboratorium. 2. Ruangan pembedahan. Ruangan ini digunakan untuk melakukan pembedahan hewan dan mengambil sampel organ tubuh dari hewan. Nah, untuk mendukung pemerintah dalam mempercepat penanganan COVID-19 di masa pandemi, ruangan di Animal BSL-3 ini dialihfungsikan untuk mendeteksi COVID-19. Sehingga, semua ruangan di Lab BSL-3 digunakan secara aktif untuk semua kegiatan deteksi dan riset SARS-CoV-2. Seluruh ruangan di BSL-2 dan BSL-3 dilengkapi dengan berbagai peralatan yang mendukung aktivitas penelitian, juga didukung oleh sumber daya manusia yang terdiri atas para peneliti dan prekayasa terlatih, tim logistik, dan tim administrasi. Melalui fasilitas laboratorium BSL-2 dan BSL-3 yang dimiliki, LIPI siap berkontribusi secara maksimal untuk mendukung pemerintah dalam mempercepat penanganan COVID-19. Sahabat LIPI dimanapun berada, jangan lengah, jangan menyerah, dan jangan lupa untuk selalu mematuhi protokol kesehatan dimanapun berada ya. Bersama, kita bisa melawan pandemi ini. Salam sehat dan semangat ya!
Good morning, distinguished guests, ladies and participants. Thank you for joining today's webinar, which is the first webinar of Enzyme series presented by Center of Excellence Integrated Biorefinery Research Center for Biotechnology LIPI. This morning, I would like to extend very warm welcome to all participants who have joined this room, and also from YouTube official LIPI, from ministries, Indonesian Institute of Sciences, Bogor Agricultural University, from Gajah Mada University, Brawijaya University, Also, good morning from Indonesia for King Mongkut's University Technology, Thonburi. I hope we always in a good health amidst this global pandemic. Ladies and gentlemen, before we initiate the event, allow me to read the rules of today's webinar. Attendees are required to using proper ID, indicated or real name and affiliations. For example, Aulia_LIPI. The attendees setting up disabled for speakers and videos, only any allowed by the operator can be able to. Attendees are welcome to ask questions via Q&A in Zoom or live chat on YouTube LIPI with the following format: name, _affiliation, _ to whom questions will be addressed, _question. Please kindly fill out the attendance form that will be shared via chat feature, and also the e-certificate will be given by email to attendees who filled the attendance form. And for the agenda for today, firstly, we will have an opening remarks conveyed by Director Research Center for Biotechnology LIPI. And then we move to the main agenda, which is plenary sessions from Enzyme Technology Laboratory Pilot Plant Development and Training Institute, and also from Novozymes Company. We move to discussion sessions with Q&A, and then we will have another plenary session by Head of Center of Excellence for Integrated Biorefinery Research Center for Biotechnology LIPI, and then followed by discussion sessions with Q&A, and we have closing. Distinguished guests, ladies and gentlemen, may I have your attentions? We would like to initiate webinar in a very few moments. The Honorable Dr. Puspita Listianti, Director of Research Center for Biotechnology LIPI. Good morning, Ibu. Dr. Engineering Nanik Rahmani, Head of Center of Excellence for Integrated Biorefinery Research Center for Biotechnology LIPI. Patra Pasan, PhD, Enzyme Technology Laboratory Pilot Plant Development and Training Institute, Thailand. And also Dr. Chen Alvin, Quang Teo, Industry Technology Specialist, Novozymes Company, Malaysia. Honorable Directors of Research Centers and Head of Bureaus in LIPI, representative from Ministry, R&D institutions, and also from universities. Distinguished moderator, journalists, media, and all participants. Good morning. It is my pleasure, Dati, to bid you all a genial welcome to the first webinar of Enzyme series, Current and Future Trends in Enzyme Technology and its Applications. This webinar is organized by Center of Excellence, Integrated Biorefineries Research Center for Biotechnology LIPI. Ladies and gentlemen, this webinar also conducted virtually live from Zoom meeting and also official YouTube channel of LIPI. And this morning, we would like to begin today's occasions with an opening remarks that will be conveyed by Director Research Center for Biotechnology LIPI. Please welcome Dr. Puspita Listianti.
Thank you, Mbak Aulia. We meet again in an international webinar. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable plenary speaker, Assistant Professor Dr. Patra Paison from King Mongkut University of Technology, Thonburi, Thailand. Honorable plenary speaker, Dr. Alvin Teo from Industry Technology Specialist from Novozymes, Malaysia. Also, Honorable plenary speaker, Dr. Nanik Rahmani, Head of Center of Excellence Integrated Biorefinery Research Center for Biotechnology LIPI, and also Head of Dr. Vina Amreta, Head of Research Group of Enzyme in Research Center for Biotechnology LIPI. Distinguished all participants from all various institutions in Indonesia, Thailand, and Malaysia. Ladies and gentlemen, good morning to all of you. It is a great honor for me to have you all in the interesting meeting webinar Enzyme series with the topic current and future trend in enzyme technology and its application. For today's webinar, I would like to report that we have received a high appreciation from 215 participants from various sectors such as government, private, and universities. This webinar series, I also would like to inform you that it will be in a series, will be held annually together enzyme expert from various countries, various sectors, and various enzyme research to share advanced and new technology in enzyme world. We hope this seminar will support the international collaboration among scientists, professors, and research professionals, also young researchers, collaboration, industrial delegates, and student communities across the globe. In this webinar enzyme series, we will discuss research, achievement, and advancement in enzyme technology and bioengineering field for the bright future. Distinguished guest participants, ladies and gentlemen, this webinar is organized by Center of Excellence, Integrated Biorefinery. That one of the focus topic is enzyme-related research, especially in lignocellulosic degrading enzyme as materials for converting biomass into valuable products such as bioethanol and lactic acid. We have 5-year collaboration, long collaboration with Kobe University, with Indonesian University, and then also Nagasaki University regarding this topic. We hope in the future we can make collaboration with other ASEAN countries such as Thailand and Malaysia. So please enjoy the webinar. We hope with the various background of participants can result good collaboration among enzyme researcher and enzyme user. To end my remark, I once again would like to express my gratitude to all committee for the hard work to prepare this webinar. Now, allow me to declare this webinar is officially open by saying Bismillahirrahmanirrahim. The first webinar enzyme series, current and future trends in enzyme technology and its application. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Ibu, for the warm opening remark this morning and to officially open this webinar. Ladies and gentlemen, before we move to the main agenda, we will have a group photo. Please kindly activate your camera. I will count on 1, 2, 3. Once again, 1, 2, 3. Thank you. Gently reminder, please kindly fill out the attendance form that has been informed by the official committee, and also please write down your name correctly for the e-certificate purposes. Distinguished guests, ladies and gentlemen, now we have arrived at the main agenda of today's webinar. In the following an hour, we will have the first plenary sessions, which present two speakers from Enzyme Technology Laboratory Pilot Plant Development Training Institute, Thailand, and also from Novozymes Company, Malaysia. There will be prize for the most active participants in these sessions, so please kindly be active during the discussions and Q&A sessions. Ladies and gentlemen, our moderator for the plenary sessions will be Dr. Vina Amreta Laksmi, researcher from Research Center from Biotechnology LIPI. Before starting the sessions, Allow me to read the brief profile of Dr. Vina. Dr. Vina is a researcher of Research Center for Biotechnology LIPI. Currently, she is the head of Enzyme Engineering and Technology for Bioproductions Research Group, and her research interests are on protein engineering, molecular microbiology, enzymology. She obtained her doctoral degree in philosophy at the Biological Science and Technology, Kagoshima University, Japan. As a researcher, she is also active in producing several publications in collaborations with international partners. Dr. Vina, please welcome, and time is yours to lead the plenary session.
Thank you very much, Maulia. Hello and welcome to today's webinar, the first webinar of Enzyme series. Today is all about uncovering current and future trends in enzyme technology and its application. Firstly, let me once more time introduce myself. My name is Vina Laksmi from Enzyme Engineering and Technology for Bioproduction Research Group, Research Center for Biotechnology LIPI, and I will be responsible for hosting this session. Today's webinar will be divided into two sessions. You will have the opportunity to submit text question to today's presenter by typing your question into the Q&A panel, and you may send in your question at any time during the presentation. We will collect this and address them during the Q&A session at the end of the presentation. Now I would like to welcome our speaker for the first presentation. Welcome, Dr. Patrapasian. We are so glad you could be here today to share your insight on today's webinar. Before the presentation begins, I would like to introduce our first distinguished presenter, Dr. Patrapasian. So Dr. Patrapasian is from the Enzyme Technology Laboratory Pilot Plant Development and Training Institute, Thailand. And her specialty is in enzyme and bacterial technology and lignocellulose valorization. And she has professional experience from 2013 until now. And now she is an assistant professor at Enzyme Technology Laboratory Pilot Plant Development and Training Institute, King Mongkut University of Technology, Thonburi. And then if anyone here interested in her publication, also can check her publication in this slide. Okay, thank you very much. All right, without any further ado, I would like to welcome Dr. Patrapasian. The time is yours.
Thank you, and good morning, everyone. My name is Patrapasian, and today it's very nice to join this first webinar because it's a good chance for me also and some of my team that can join and opening our collaboration. And thank you again for a very nice warm welcome from the Indonesia. And I think in the future, we also can have a chance to get more information between us. And also, good morning from Thailand to Indonesia and Malaysia also. So I would like to start my presentation today. So let me share my slides from the screening. Okay, could you see my slide clearly?
Yes.
Okay, so good morning again. And my presentation, I will give some more information about my research field. It's about the lignocellulose degrading enzymes. It's the potential biocatalyst for adding the value of local bioresources. And I think many keywords will be related and familiar with me and all of you, because I know today all of you are the same focus about enzymes and how can we apply and use it for a very high value. And again, my name is Patrapason. I'm the researcher at the Excellence Center of Enzymes Technology and Microbial Utilization. And I'm working under the Pilot Plant Development and Training Institute at the Kin Mongkut University of Technology, Thonburi. Of course, you can visit me in Thailand, and this is my university. And of course, now it's pandemic, so normally we work from home. But hopefully, after the situation better, hopefully some of you can visit and we can share some more environment and knowledge together. And what is about the enzymes technology that we try to driving our enzymes? Of course, it's about lignocellulose. And the lignocellulose is a lot in our country. We would like to make the agricultural residue or the waste to the high value product. Of course, this is the bio-based, but we want to do our technology by applying enzymes and microbial technology to make it more useful and sustainable. And of course, after we develop our technology, the enzymes can be a biocatalyst to change the agricultural residue and generate high value added for apply in the food, feed, fuel, and also biochemical. And today I will show you some of our research that we try to apply our enzymes. And of course, lignocellulose is one that we interest because I think in our country, we have a lot of agricultural residue. For example, rice straw, palm residue, or some corn stover or corn cob. That is the byproduct after we harvest it or we do the some food processing. For example, bagasse is also come from the sugarcane factory also. That's why you will see that the lignocellulose is abundant. We can have many places in Thailand, Indonesia, and all over the world. And of course, it's cheap and sustainable. That's why if we can use it with the very effective way, we can make the very high value with the environmental friendly technology. And this figure, I would like to show you about the structure of lignocellulosic biomass. Of course, the major plant is the cell wall that is containing three major components. It is cellulose in the green color, hemicellulose in the purple, and the lignin. And normally, these three components will link together and make a very complex. That's why if we would like to use each of them, we need to degrading into the fermentable sugar. And for example, cellulose, if you use the technology to degrade cellulose completely, you will get the cellulose. And one technology that very safe and good for environmental, this is enzymatic hydrolysis. And you will see that after you degrade the cellulose completely, you will get the glucose as the major fermentable sugar. And of course, this glucose is the building block that you can ferment to a lot of high value added products. For example, ethanol or some sugar, lactic acid or chemical. And not only cellulose, hemicellulose is another polymer in the biomass that you can degrade them to be a monomer. The majority is xylose. And of course, this sugar also can continue ferment to another high value product like ethanol or some xylitol also. And nowadays, not only cellulose and hemicellulose, lignin is also a new trend that if we can generate the lignin and its derivative, of course, it will be a good biochemical that will be trendy. And nowadays, lignin value and price is also increased. But technology is what we need to develop, and we need to looking for an effective way to generate this lignocellulosic biomass to be a very high value because we have it already, this lignocellulose, but we need to learn and study how to make it very nice and beautiful way to generate a very good product. And of course, we need the enzymes to generate the high value added product. And I will tell you some more example about how we can generate the enzyme and develop them to produce the high value. This is the key technology. If you would like to use the enzymes, this figure shows the plant cell wall. You see that the plant cell wall is also show the very complex structure that I told you, that cellulose, hemicellulose, and lignin. There will be a good friend. That why they stay together very close. But however, if we need to use it, we need to saccharify all of them to make a fermentable sugar. And the key enzyme that need for cellulose degrading, you can see in figure A, for cellulose degrading, it needs a lot of enzyme sets. The major is endoglucanate to cleavage the cellulose chain. And not only endoglucanate, for completely degrading cellulose, we still need another enzymes, for example, exoglucanate and beta-glucosidase to make the synergistic action and degrade all of the cellulose fragment to completely hydrolyze it and get the glucose. And another way for silane degrading in the figure B, you can see that silane is very complex structure because it has the silane chain that modify with the branch chain also. And for silane degrading, you see that it's a lot of enzyme needed for silane degrading. Of course, the key chain of the silane is the cellulose linked by the glycosidic bond. Endosilanase is the key enzyme for this work that they can randomly cut down the silane to generate the oligosaccharide. But from this picture, you will see that a lot of enzymes also need to cleavage the silane. It's, for example, the branch chain of the silane. It's decorated by a lot of sugar. For example, arabinose sugar, glucuronate, galactosidase also need to remove some branch chain also. That's why for silane degrading, you see that a lot of enzyme also need to generate together to degrade the branch chain and also degrade the silane chain of the hemicellulose. And from this figure, I want to address that not only key enzymes need for the saccharification, we also need another accessory enzymes. For example, expansin, swollenin, and especially lipid polysaccharide monooxygenase or LPMO. This also the helping enzyme. Of course, it's not directly active to the glycoside hydrolase, same as the key enzyme, but it will help the to degrade and change the structure from the crystalline to be amorphous. It means the structure of cellulose will be more loosened and easily. It will help for opening the structure and make the key enzyme working effectively. And another way that our laboratory focusing is about multifunctional enzymes. It means one protein have many activities of enzymes. This enzyme that we discover, it means we can save time to produce the enzyme. One enzyme that have variety action. And this is some paper that we published and try to apply this enzyme for saccharification by all pretreatment process also. And from the way that you know why enzyme is important, what we do in Thailand, it means we try to looking for an effective enzymes from the natural, because we know that in our country, the tropical area have a very diversity of the microbial and enzymes. That's why we have a very good source for looking for the very good microorganism and the enzymes. And of course, nowadays in the commercial, mostly enzyme produced is from fungal. That's why we would like to generate another chance for the enzyme technology by develop the enzyme from bacteria, because we know that bacteria mostly they can tolerate and survival in a very harsh condition. They can tolerate in more extreme condition like a temperature or more stronger under very broad pH. That's why it's a good source of the enzymes that we can looking for the effective one. And what we try, it means we do the screening. Of course, it's from the aerobic condition and under an aerobic condition. And after that, we will identify the bacteria and the enzymes to know the property of them. And after that, not only understand what is the property of the enzyme, we also try to production and characterization, how to make it working well, even in the lab scale and also some pilot scale. Because when we produce the enzyme, sometimes we can see the very good in lab scale, but also we have to link between lab to commercial also to push our enzyme and launch in the real market. And this figure show you the easy way what we do about enzyme screening. Try to looking for the unique enzyme property that we focus about biomass degrading. If the enzymes can degrade biomass, the complex structure will change to the sugar under the saccharification process. And from now on, the sugar can be fermented to the high value added product. And I know that some of you will be understand this biotechnology way that we can generate the very high value added from the enzymes technology. And here, this is some example of our enzymes and culture collection. We also have the enzyme from aerobic bacteria and anaerobic bacteria. Of course, each of them have the unique property. And now we try to develop our enzymes to use in many applications. For example, the first enzyme that from aerobic bacteria, we publish about the knowledge, and also we try to develop these enzymes for extraction. This enzyme is named multi-enzyme complex. How can we know the multi-enzyme complex? Because we screening the bacteria that can degrade the lignocellulosic biomass without pretreatment. That's why the structure of biomass is very rigid. But this bacteria also can grow and secrete the effective enzyme. And the pattern of enzyme will show you in the SDS page and enzymogram. You can see that it's a lot of enzymes subunit containing a lot of silanase and cellulase. It means that this crude enzyme is available for degrading plant cell wall. And that's why we try to apply to extract the beta-glucan from the mushroom, because we know that the mushroom also containing cellulose and hemicellulose, not only glucan containing. And after we put our enzyme from Penibacillus, it helps to generate the beta-glucan extraction. That is one of the example of the enzyme application. And not only aerobic bacteria, we also can generate and screening for anaerobic bacteria also. In this anaerobic bacteria, we benchmark with the very high complex enzyme system, Clostridium thermocelum. That is our benchmark. We're looking for the bacteria that can degrade crystallized cellulose by decrease the turbidity faster than the benchmark. And we looking for the enzymes property of this strain. We know that this strand is Clostridium thermocellum S14. That is very faster degrade crystallized cellulose. And this is the structure of the bacteria cells. And after you apply this enzyme from the bacteria, comparing to the fungal in the blue line to degrading rhizole, what it happens when we study about the rhizole degrading, you will see that after enzyme pretreatment, you can see the structure of rhizole was destroyed. And you see that glucan digestibility of the cellulosome is the highest, more than 90%, when we make the cellulosome, our cellulosome cocktail with the beta glucosidase action. It will be better than the fungal complex system. And this is some informative knowledge about the complex enzymes that we know about cellulosome system. And we try to apply this system to generate the high glucose digestibility. And this is some of our enzymes collection. We have a lot of enzymes that try to screen using the unique property that it can degrade the substrate specific. For example, cellulase, hemicellulase, multifunctional enzyme, and also some manganate and LPMO. Of course, each enzyme, it has some specific character, and we study individual enzymes and try to mix them together because each of substrate have a different complex structure. And here, it's one of our multifunctional enzymes that I would like to show you some importance of these enzymes. The name of the enzyme is glycoside hydrolase family 43. This enzyme it's specific for silane degrading. It can degrade birchwood in figure 1A. You see that after you put the enzyme to the silane, it can randomly degrade silane to be a silooligosaccharide. And interestingly, it can generate xylose also because this enzyme has actually 3 functions. It has the function of endosolanase to randomly degrade silane. And of course, we have the specific substrate. It's short chain arabinosilane. That's why in the figure B, after we generate the specific substrate with our enzyme, the enzyme can degrade the short chain arabinosilane to be xylose and arabinose. So it means this enzyme can digest the short chain arabinosilane and generate completely the xylose and arabinose. And also, when we try to analyze the beta psilocybate activity in the figure 2, you will see the pattern of the TLC. And you see that after you put the enzyme with the xylose, you can degrade xylose, xylobiose to get the xylose as the final product. What I would like to telling this information, it means the enzyme can degrade silane, that very complex structure, and generate the xylose as a major product. Because nowadays, we need a lot of enzymes. As I show you that we need at least 3 enzymes to help each other to completely degrading the silane to get the xylose. But this enzyme is a unique property. It can adapt themselves, preventing the silane to get the finally xylose as a major final product. What is the good of this enzyme? When we generate just only 1 enzyme with the 3 activity, it means we can save time and save the process for enzyme production, and we can generate the effective one. It means the step will be more easy, and the technology also simplify also. And what we try to apply this enzyme, it also can use for biological for xylose production. As I show you that this enzyme can cleavage silane and generate xylose yield. And from this figure, you will see that in native form of the biomass, they cover with the hemicellulose and cellulose is inside. If you can put the silanase, it will be remove some hemicellulose like a peeling of the lignocellulosic. And when you see the cellulose yield in the figure 3, comparing between untreated rhizole in the blue bar and the ammonium-treated rhizole in the red bar, you will see that the our dry functional enzyme PC AXY33A is very effective. Even the untreated and treated rhizole, you can generate cellulose yield more than 60%. It means without pretreatment by ammonia, you also can get a very effective action of the enzyme. Comparing to another truly silanase, what is truly silanase? It means normally silanase is containing in glycoside hydrolase family 10 and 11. But after we try to put the silanase 11 and family 10, it seems very low cellulose yield generate because this enzyme is the endosilanase. It will cleavate randomly and generate oligosaccharide, not completely cellulose degrading. That's why if you need cellulose, this enzyme is very good for cellulose production. And when you're looking for the structure of enzyme, this enzyme GH43, also they have some CBM family 6. CBM is another domain of the enzymes that we interest in this point because it can, CBM is a carbohydrate binding domain. It can bind very close with the substrate and it will bring the enzyme and substrate very close and easy to degrade. That's why if normally when enzyme containing CBM, it will help the enzyme to degrade insoluble substrate better than without CBM. But however, this GH43 with CBM, they can generate cellulose better than GH10 without CBM and GH11 with CBM also. This is also proof that if we looking for the enzyme's property for cellulose yield production, this also a very good choice for cellulose production. And of course, this enzyme is specific for arabinoxylan degrading. And we also try to apply this enzyme for the animal feed, because you know that in animal feed, the component of animal feed mostly is from cereal, and the cereal component containing arabinoxylan. And this arabinoxylan is the problem because it will make the animal feed with the high viscosity, and the high viscosity will also trap the nutrient from the feed, and then the animal is difficult to digest and absorb the nutrient. So after we add this enzyme, it seems that our enzyme can breaking down the arabinoxylan that containing in the plant cell wall or the cereal cell wall, and after that the nutrient is easy to absorbed by the animal, because after the arabinoxylan was degraded, the viscosity was reduced, and that's why it's easy for digest by the animal itself. That's why this enzyme also is a good trend for animal feed also. And nowadays, I think that a lot of animal feed, we need to replace the antibiotics. And of course, most of enzyme is trendy to apply in the animal feed. It can reduce viscosity, as I told you in the previous slide, and also the product of enzyme degrading. It will generate some short chain oligosaccharide, for example, oligosaccharide, that it will be, we know it's in terms of the prebiotic, that is XOS or the short chain oligosaccharide, it will be good for animal health also. And this slide, I would like to mention again, when we use the enzymes technology, comparing to another chemical or mechanical technology, you will see that the enzymes technology is a specific action. You can put the specific enzyme that will target to your substrate. So the action, it will be specific, and my reaction, because it just use the buffer, and even you put the base condition, you will get high yield, and of course late contamination. And the important is mean non-toxic if you use the enzymes. Comparing to chemical or the mechanical, you can get some low yield, and of course the some impurity occur in your process. That's why we know that enzyme is also very famous, and that we try to looking for the effective one and try to make it more active in the condition also. And another application of the enzymes, you know that mostly the enzymes can help us to degrade the high molecule to be a short chain oligo or the sugar. This is the platform of the enzymes that we can do for the pretreatment, saccharification, and fermentation. Mostly in Thailand, in some season, and I know some in some country also, after they harvest the corn stover, mostly they burn the corn stover and make the pollution. That's why we need to looking for another technology to replace the burning. We have to looking for the sustainable technology to stop the toxic and the environmental problem. And what today we discuss is about enzymes, right? Because enzymes, it's a very good bioprocess technology, and we can generate biorefinery. And if we're looking for this platform, pretreatment also needed, because the structure of lignocellulose is very rigid and difficult to degrade. And of course, pretreatment, we have a lot of pretreatment, like a chemical, physical, and also biological pretreatment process. This is the bottleneck, because if you can do some good pretreatment, you can get the easy step for saccharification to get the high yield of fermentable sugar, and you can generate the good fermentation product like the ethanol, acetic, and succinic. So another bottleneck is pretreatment. And what I would like to show you today is the biological pretreatment that also you can apply the enzyme for this purpose also. And if you see, this is just the update result and paper that we published in recently about biological pretreatment of the corn cob. Corn cob also is a byproduct of the corn process. And what we try to do, we're looking for the effective enzymes from Bacillus K1. What is the specific of this enzyme? These enzymes can produce silanase without cellulase. It means good for biological pretreatment because it will target only silane. And then you will remaining a lot of cellulose that will not damage. And you can use cellulose for another purpose effectively. And this technology, we use Bacillus K1 bacteria put into the corn cob. And then this technology is also cheap because corn cob is a waste and it's a low price, and we can generate a very short time pretreatment. It's like a 12-hour and low. This bacteria and enzyme, it's low lignin absorption. That's why no need chemical pretreatment. Just put the bacteria and it will do the biological catalyst by itself. Let's see in the figure one. Figure one show you what happened after we culture the bacteria with the corn cob. Of course, we are monitoring the enzymes, that is cellulanolytic enzyme without cellulase, and this enzyme generates very fast, whether 12 hours. You can see in the green line, cellulanolytic enzyme will generate very fast during 12 hours, and after that, they can produce some reducing sugar also at the 6 hours, and after that, some sugar was consumed by bacteria, and finally, you will generate the enzyme in the solution at 12 hours. And let's see what happened in this fermentation. You can pretreat corn cob. That at the same time, you will produce the enzyme in your fermenter, and the residue in the solution, you will get the silooligosaccharide. This is the sugar pattern or oligosaccharide pattern in the solution. After you separate the enzyme, you will get the oligosaccharide at the 6 hours. You will see the corn cob silane was degrading in the short chain of oligosaccharide, and you will see the product from X1 to X6. It means variety of product at the 6 hours. And after that, when the bacteria grows at 12 hours, it will consume some short chain oligos sugar like a X3, X2, X1. They will eating, and then finally, the final product is only major silooligosaccharide. It means X5 and X6 only. And let's see this technology for biological pretreatment. You will also get byproduct. It means enzymes that can apply for another application. And then silooligosaccharide you will get from this process, that it will be a good prebiotic. And after that, the corn cob, after biological pretreatment, we see that the structure is very structure completely changed. When you see the SEM structure, you see the silane was peeling. That's why when you apply the commercial cellulase for further saccharification, in this research, we try to add the two types of commercial enzyme. It means acrylase and CTEC2, comparing between untreated in the blue line and treated one with the green bar. You will see that when the silane was removed by our biological pretreatment, the commercial cellulose can easily attach to the cellulose and generate glucose from this corn cob higher than unpretreated. And one interesting, it means when you use biological pretreatment, you generate the low toxic. That's why all of the enzymes will be applied in the saccharification. It will generate some toxic or inhibition. That's another key interest when you apply the enzyme for biological pretreatment. And this is just some of the application when you use the bacteria to secrete the enzyme and use for biological pretreatment. But this is just the beginning. We also try to develop more by use enzyme for agricultural residue valorization. And not only corn cob is like a model for the beginning, but the technology also try to develop to generate the biological pretreatment. As I told you, you can generate the silanase. That of course it can apply in animal feed or some bakery and some pulp and paper industry. That it's good for silanase can working well. And after you do biological pretreatment, you will get another byproduct, that is silooligosaccharide. This is the prebiotic that's useful for health even for the animal and for our health also. Prebiotic is very useful. And after that, when we do biological pretreatment with this technology, you will get the cellulose that also you can develop cellulose for more application, like to produce some cellulose packaging, to develop the cellulose derivative, or do some chemical interaction to generate the hydrogel. And or another way, if you completely hydrolyze the cellulose, this will be glucose that you can ferment it for very high value added product like ethanol or acetic acid by the yeast or some acetic acid bacteria. And another technology that we need to develop the enzyme for the interest substrate is lignin. That our lab also try to looking for the bacteria and enzymes to generate the biocompound from lignin also, because we know that lignin is not only a waste, but if we modify the lignin, it will be a good component and it will be good for the future also. That's why from these pictures, we still have a lot of work to do and study. But however, we will try our best to develop our enzymes and modify it for specific to each purpose in the future also. And this is our viewpoint when we would like to study about enzymes. Of course, we know that the enzymes it help us to break down the cellulose to generate the fermentable sugar. And it's very good for environment, and it's a sustainable technology. And what we have to do about enzymes, we need to looking for the effective way to apply the enzyme by use the low cost for the enzyme production, low cost for the process. And we need to generate the high recombinant enzyme, study the way to get the expression level. And the important is also the formulation of enzymes like the enzyme cocktail to get the very high yield and high efficiency. And not only enzyme itself, what we try to do is also about the degrading lignocellulose. Concob is just one example. And another lignocellulose that a lot in our country is the cassava pulp that I would like to show you some of our enzymes and bacteria can generate energy, food, or biomaterial from this cassava pulp by the enzymes technology platform. In Thailand, we have a lot of agricultural residue, and mostly it's cassava factory all of part in Thailand, and increasing day by day. And then that's why we generate a lot of cassava waste in the cassava factory. And of course, this waste we can use as a consume animal feed, and some is the cassava pellet and cassava starch, but the remaining is the cassava pulp that some season it's very terrible for the factory because it will generate the very bad pollution. That's why we try to help the company looking for the better way to improve this cassava pulp. First, an easy way, it means we try to generate energy by this cassava pulp. Of course, mostly biohydrogen or biomethane could generate by the wastewater treatment technology. But if you use the pulp, it's very difficult because it contains fiber, and the acidogenesis and methanogen, it's difficult to use the fiber. That's why our enzyme is the key for this technology. What we try to do is mean we screening the effective bacteria that generate the enzyme set in terms of amylase, cellulase, and cellulase also, because we know that this structure of cassava pulp, the starch was covered with the lignocellulose. That's why this cellulose and cellulose also important to degrade the fiber to be soluble. And then what we do in our lab, we screening and selective the good bacteria that can generate hydrogen from the cassava pulp. And from the first screen, we observe a very high bubble, that it means possibility to degrade well and generate a gas. And then we observe the bacteria community and study the effect of the pH and condition for hydrogen production and the concentration of the cassava pulp also to generate the better way to produce the hydrogen directly from the cassava pulp. And of course, we can looking for the interest candidate. Some of them that can degrade the cassava pulp effectively, more than 60%. And all of them can produce the enzyme like amylase, cellulase, and cellulase. And this candidate, BK16, is the best choice for us because it's got 3 sets of enzymes completely. And we proved that this bacteria consortium also can generate high hydrogen production also. And not only for the biohydrogen, the bacteria and enzyme also can apply in the food and feed. When you use the cassava pulp mixed with the bacteria and enzymes, of course, you can make the cassava pulp easy for animal digestion. And the bacteria itself also can generate the enzyme that's good for digestive of the animal. And they also can generate some oligosaccharide in the cassava pulp fermentation, and you can increase the immune of the animal also. And the product will be reduced and replaced of the use of antibiotics. And this is some of the example when you apply also the enzyme and bacteria with the cassava pulp. It means not only biofuel you can generate for the food and feed also. So the key, it means the enzyme that will match with the application that you need. And this is another application. Not only I show you the previous slide, but nowadays in our lab, we also develop the trendy and good enzyme for serving in many applications. For example, in the prebiotic, as you know, we can degrade the polysaccharide to be an oligosaccharide. For example, silo-oligosaccharide, mono-oligosaccharide, and all isomoto-oligosaccharide. This oligosaccharide is good for prebiotic because our gut microbiome is a very good food for them. That's why it will boost our immune and good for health. And another way is the rare sugar. That is means we can looking for some good enzymes that can change the structure of sugar to be a very low calorie and good for health also. And another experiment of enzyme also we can use for feed and also feed supplement, as I telling you in the previous slide. And what we try to do is mean develop the enzymes technology, change the biomass to be a high value added product. And of course, they have many steps. And if we
I'm
can
sorry
do
to disturb you, Dr. Patra.
Yes?
I'm sorry to disturb you. We have 5 minutes more until you can finish your presentation.
Okay, thank you. Thank you for reminding me. And this is just some few slides remaining about the perspective. That is the key that we try to develop our enzymes with the effective bacteria complex. We study about the genome of our bacteria that we interest, even in the very extreme thermophilic. We looking for the halophilic bacteria also to generate the bioactive compound from the bacteria production. That's why this knowledge, we also looking for the various type of lignocellulosic biomass to generate the good pretreatment or very best saccharification process. And in our team, of course, we have the pilot plant. That's why we still need the downstream process to get the effective process to produce our enzymes in the commercial scale with the high yield, low cost. And of course, one more important, it means every part needs to be used, and we focus about zero waste also. And this technology, we would like to make it simplified and generate the enzymes technology to produce biofuel, biomaterial, and biochemical. And this is some partner in inside Thailand and many countries from the in our world, and partner for collaboration, because I know that the achievement is not only in my team. We still need the partner to improve our collaboration. And this is also some international collaboration. Also, we still have some partner from Japan, America, and also from University of Alanka also, and from some part of China, that we try to develop the technology of enzymes and apply it for the many applications. And last but not least, I also invite all of you to my next seminar. If you interest, you can looking for more information about Asia bioenergy and bioeconomy, because our team also organized this meeting. We can open more about the enzymes technology under the bioeconomy and bioenergy also. And we also have some interest speaker that I think this time you build a bridge for connect my team and your team also. And hopefully in the future, we can do more and more application and innovation together. And finally, I would like to thank you again for your nice collaboration and very good chance for me and my team to exchange our knowledge. And finally, I would like to thank you, our team also, because not only me to get the good achievement, thank you, my team, to love about enzyme, encourage the knowledge and study hard and work hard to do the very effective enzyme. And thank you all of you today for your attention. Thank you and ขอบคุณค่ะ
Thank you very much, Dr. Patrapason. I hope you
Okay.
all, yes, I hope you all enjoyed this amazing presentation. Now, Dr. Patrapason will answer some questions that were sent to us during the presentation. As a reminder, you can still submit your question through your chat panel as long as we still have the time. Okay, we have around 15 minutes in this Q&A session. So the first question is from Ms. Nurul. So I'm wondering, what was your main strategy in finding the selective isolate as you targeted the enzyme producer from the bacterial community? And what was the major difference between aerobic and anaerobic bacterial screening in terms of the bottleneck? Time is yours, Dr. Patrapason.
Okay, thank you for your question. That is also the key for the enzymes technology. Also, firstly, I would like to mention about bacteria community. That bacteria is also interest in terms of it can tolerate and survive in a very different condition. You can see the bacteria in a very high temperature area or in a very strong acid tolerant. That it means bacteria can survive. And when it can survive, it means it can make the good community and the enzymes that can important for them to survival. And the another question that you asking about the difference between aerobic and anaerobic bacteria. Of course, both of them it have some unique properties. For the aerobic bacteria, of course, the technology for production maybe it's easy in terms of the condition for growth the aerobic bacteria, because anaerobic bacteria you need to do under the anaerobic condition. You need some anaerobic bench to screening. That's why anaerobic bacteria is some trick that you need to adding for the difficult for screening. But aerobic and anaerobic bacteria, they can generate the good enzymes also depend on the material or the carbon source that you feed them. And mostly the bottleneck, it means you have to think about the way for screening. That it means the both of them have some trick and key. So, but in my side, most of them is not so difficult, depend on the way that you try to focus. If you focus sharp, it means you can save your time for screening, and then you can eliminate the way to screening, and you can get the best choice of your bacteria also. Yes, thank you.
Thank you, Dr. Pataprasan, for the answer. I think it's quite clear. And then we move to second question from Mr. Harto Widodo. So how can we select and culture a certain microbiota, especially bacteria, which produce a special enzyme that possesses cellulose or lignin or cellulose degrade activity?
Okay, thank you. Mostly if you focus about the enzymes, it means you have to looking for the source of the material that contain the substrate of that enzyme also. It means if you want to propose the enzyme for cellulose, lignin, and cellulose degrading, mostly this biomass you can looking for from the natural, I mean, in the landfill also, they have a lot of leaf and a lot of agricultural residue in the soil and sediment. You also can take a chance to get that sample also. And another interesting source to select bacteria is from the agricultural factory, because they have many year accumulation of the waste, and it means the bacteria also develop and generate many circle to degrade that lignocellulosic biomass. And for the screening, if you would like to get the best bacteria, it means try to do the substrate without pretreatment. It means you will get the very natural and very difficult substrate. And if you can get the bacteria, it means you will get the very good microbiota that will be tolerant with the harsh condition. That also the key for the looking for the good screening and selection also.
Thank you, Dr. Patraprasan. I hope it helps. We move to third question from Ms. Fauziatul Fitria. So the question quite long. Thank you for your nice presentation. Currently, I have experiment on the utilization of white rot fungi for degrading several ways, such as empty fruit bunches of oil palm into feedstock. We have several problems to do scale-up direct fermentation from the fungi, so I'm considering enzymatic hydrolysis now. For scale-up feedstock production using lignocellulosic enzyme, can you share some of the challenges and compare them with direct fungal fermentation?
Okay, thank you for for your question, and thank you for sharing your experience with me. Of course, in my experience, mostly I'm working with the bacteria, but I know that fungal, they still have some technique that mostly for the scale up. I think the key, even for the fungal or bacteria, the key for the scale up, it means your starter. If you can generate a very active and fresh starter, you will can get a very good starter material even from fungal, and then you can put into your fermenter. And I think this good starter will easy for scaling up. And for scaling up part, the condition also important. You need to monitoring about pH, temperature, everything that your fungal love, you have to push them and spoil them. And then I think your bacteria and your fungal will growing well and decrease the enzyme higher. And another way is inducer. If you know about your fungal, maybe you have to think about what they like or something that will induce your enzyme. For example, if you culture the fungal with the sugar and you culture your fungal with the fiber or substrate, that's difficult to degrade. The sugar, it means just only growth, but they will not induce some enzymes that you need. But if you culture into the fiber or some polysaccharide, it will help for induce your enzyme production. That is another way that you need to think about scale up. And if you think about the some fermentation process, maybe from mostly in bacteria we use submerged fermentation. It means a lot of water. I don't know, in your fungal maybe you use some semi-solid state, something like that. Maybe it's also challenge for the fermentation technique. That it means maybe for the substrate loading or carbon source loading is also one challenge to produce the bacteria and enzymes also. That is many possibility and many techniques also. So maybe case by case that I think you should try to develop, and hopefully finally you will get a good fermentation and good result for your enzyme. And or if any information that I will help, you can contact me, and I'm happy to collaboration and help you if I can. Thank you.
Okay, thank you for your insightful advice, Dr. Patra Patience. We can move to the next question maybe from Miss or Mr. Sona Suhartana. There are two questions. The first question is, can Bacillus Furnus K1 cellulase, free selenolytic enzyme, and commercial cellulase be applied in the forestry sector, especially by HTI, industrial plantation forest, and HPH, forest tenure rights holders? And the second, maybe first question first.
Okay, I'm answering for this first question. It depends on your process also. Firstly, we use the cellulase-free selenolytic enzymes because we want to focus only selenase. So it means specific only selenase, and the remaining is been majority is cellulose, and your final product will be a major is the glucose. Then you can link with the glucose fermentation process, and of course some commercial, they will mix the enzyme between cellulase and selenase. You can use it also, but the final product, you will get both of glucose and xylose for fermentable sugar. So you have to think for the next fermentation step that you have to generate the yeast or the bacteria that can consume both of xylose and glucose sugar. So both of these technology have the advantage and disadvantage also. But what we try to propose, it means only cellulose we focus, and then we try to remove cellulose first. It's like a fractionation, and after that you can use cellulose for the further step. So the important, it means the next application that you need also.
Okay, thank you very much, Dr. Patrapasian. For second question, already finished? For second question,
For the second,
has there been any research related to that, related to the first question, and how does it contribute to Indonesian forestry sector in particular?
I think enzyme it's useful for many applications, but what we need to develop, it's been fine-tuned for the condition. I think mostly cellulose-free cellulose is also can apply, but we have to make some consider about condition, what is the pH and what is the temperature also. But I think it have the possibility to do in the future. Thank you.
Thank you very much, Dr. Patrapasian. Hopefully your answer can give insights for the development in the forestry sector in Indonesia. And unfortunately, the time is run out for the Q&A session. And once more time, thank you very much, Dr. Patrapasian, for coming today and share your knowledge with us. Hopefully, we can gain so much and apply it in our research or our interests. Thank you very much. Okay, we move to the next session or give back to the MC, Mbak Alia.
We will have break for 5 minutes, Dr. Vina.
Okay, thank you very much.
Thank you, Dr. Vina, to lead the first session, and thank you to all speakers for the informative informations for the sessions. Distinguished guests, ladies and gentlemen, we will have break for 5 minutes before having the next plenary sessions this afternoon. Please stay in the room and don't leave the room. Thank you. Distinguished guests, ladies and participants, we will continue to the next agenda. This morning, we will have the second plenary sessions that will be conveyed by Head of Center of Excellence for Integrated Biorefinery Research Center for Biotechnology LIPI, Dr. Engineering Nani Rahmani. And for the sessions, may I kindly invite again Dr. Vina to lead the second plenary sessions. Dr. Vina, screen is yours.
Thank you very much, Mbak Lia. I'm sorry, the schedule is for the presenter, I mean, Dr. Alvin, right?
We apologize for this, Dr. Alvin. Please have the sessions with Dr. Vina this morning.
Okay, thank you very much. Welcome, Dr. Alvin Siketeo. How I call you?
You can call me Alvin. Selamat pagi.
Oh, okay. Thank you very much, Dr. Alvin. Selamat pagi. So before the second presentation begins, I would like to introduce our distinguished presenter, Dr. Alvin. So Dr. Alvin Chen Kwang Teo, PhD, is from Industry Technology Specialist at Novozymes, Malaysia. And he finished his doctoral degree in Analytical Science, University of Warwick, UK, University of Birmingham, and Aston University, a secondment. And his research is about bacterial membrane protein as potential antibiotics. And he has experience in the application scientist with over 12 years of R&D and TS experience in household care, personal care, and healthcare related fields. And anyone who interested in his publication can check out here. Okay, thank you. And all right, without any further ado, I would like to welcome Dr. Alvin. Time is yours.
Thank you very much. I realized I didn't use the same background as you, but I hope you like this nice and green.
No
morning.
problem.
Yeah, it's actually the same weather here in Kuala Lumpur. I'm based in Kuala Lumpur now. It's nice and sunny. So without further ado, let me share my screen. Let me know if you can see my presentation. Can you all see my presentation?
Not yet.
Not yet. Try again. Sorry, not very good with didn't click share. Sorry about that. Technical errors.
No problem.
Can you see my screen now?
Yes. No, it's okay.
Okay. So let me start. So thank you so much for the opportunity for this talk, and thank you so much for inviting me as the industry speaker. So I'm Alvin. I'm based in Kuala Lumpur. I'm a application or technical specialist, if you call it, supporting detergent manufacturers across Southeast Asia to adopt hopefully more and more biotechnology into consumer products. And today, this morning, say within 30 minutes or so, I would like to bring our attention to the application or the use of enzyme, of the power enzyme, focusing on the household care industry. Next, you can still see my screen, yeah?
Yes, no problem.
Cool. Corporate slides. Let me introduce you a bit to Novozymes in a nutshell. So Novozymes is a global biotech company. We are the market leader in industrial enzyme of over half of the global market share. The headquarters is in Bagsværd in Denmark. We have just around 600 plus employees. Over the years, since the beginning of Novozymes, there's over 700 products being developed to be used in 30 plus industry across 140 countries in the world. We have over the years 6,000 plus patents, still counting, And just last year itself, there's 18 new products being launched in these various industry. Novozymes invests about 13 to 14% of our revenue back into R&D activities per year. And that's something we're very proud of, to continuously drive innovation in this field of biotechnology. The purpose of the company is really to, together, we find biological answers. In this case, today, I'll focus the biological answers as enzymes. For better lives in a growing world, the world is ever changing, and we constantly ask ourselves in our daily work, how can we rethink tomorrow to bring about a more sustainable future for humankind? Right, the biological answers, like I alluded, comes from enzymes or microbes, and we heard some fantastic from Dr. Pason just now. Think about it, this morning, maybe some of you have roti canai for breakfast, or some bubur, some porridge, right? When you start chewing the roti in your mouth, then your mouth start producing saliva, and saliva have amylase. So to one of the enzyme classes then start digesting the food besides the mechanical action of chewing. It goes to your stomach. And then you have more enzyme comes into the picture. You have amylase, you have protease and lipase to help really digest the food so you can absorb this organic nutrient and then you can sustain your growth. It's exactly the same principle we employ this one in the cleaning industry in household care. But enzymes are also used in other industry like Dr. Pason just alluded in. And you can look at this picture, hopefully you can find some similarities here in your daily life. Say for instance, dirty pairs of trousers. Before COVID, maybe we go out for a football game or basketball, or jogging, hiking. You come back with a dirty pair of trousers, right? Then you need to wash it. Detergents contains enzyme. Or in food and beverages, say lactose-free milk, or yogurt, and things like that, enzymes can be applied, for better or different nutritional products. Or in the field of biofuel or in animal feed. The various industry, that's why our enzyme application are covered in so many industries across the world. And enzymes can be applied in either the finished product, like detergent contains enzyme as the finished product, or it can be used to as part of the process to make a product, say in the beverage industry, or it can be used as a key ingredient to increase the yield of a particular plant process. So there's a different versatility of the enzymes to be applied in the industry. And I don't think this slide is something that is unfamiliar to us, but let me draw attention. Enzymes are just strings of amino acids, right, folded in the right 3D structure. In terms of household care, we are focusing on the globular proteins, so water-soluble proteins, different classes. And one of the key value proposition for enzymes in the household care industry is enzymes are nature-derived. They come from nature. So you can think of a lot of exotic trips across the world to find the new microbes, bacterial, fungi, do your sequencing, do your bioinformatics to find new gene sequences that potentially can turn into target molecules for exciting application. Then many rounds of screening, testing the sort of purified enzyme in different types of application, multiple rounds. And then with all this data generated, how do we then further test again, build models, or encourage a cross-industry fertilization in terms of the usage of particular classes of enzymes. Enzymes are biocatalysts. This is not something strange to us. It drives chemical reaction multiple times, lower the activation energy. And with some unique characteristic of this biocatalyst in contrast to conventional catalysts or chemical-based one, I want to bring you to attention in subsequent slides that we believe that with more and more R&D effort, we can show that enzyme can effectively replace the chemicals. And what I mean by chemicals are not the biochemicals, are the more traditional synthetic or petrol-based derived chemicals to reformulate into a more sustainable detergent product. And Novozymes, being the world leader, we believe that we are uniquely positioned to drive innovation in this industry to benefit humankind and the environment. And that sort of resonates in how we always want to create a bigger and bigger libraries, so to have more and more genome sequence, find the new strains of microbes, to lead to better answers in terms of application. And also, how do we shorten the development pipeline? So typically in R&D of upstream process takes 5 years to really find a good potential molecule then to be developed into a product. How can we shorten into 3 years so that then we can bring a good product into market faster? And then just now Dr. Paso mentioned that when you have good candidates, right, how do we then optimize production, fermentation, getting that kind of economy of skills, optimize the different processes, fermentation down to purification, and then formulation. So enzyme, we don't self-purify enzyme per se, not just the enzyme molecule. Is embedded in a matrix of different ingredients to stabilize them for the end usage in the plant or in a detergent product. And then having good products itself, we also need to have diversified global presence. So we basically have footprint across the world. We have a few major R&D centers in Denmark, in China, in the States, but we also have a lot of commercial offices around the world. Like myself, I'm based in Kuala Lumpur. And that is really to deploy and to help the local customer adopt a developed product and then help the daily troubleshooting, for instance, or help them to make new products. And that's what we believe we are bringing into the biotechnology market. In terms of enzyme discovery, like I mentioned just now, so we are always looking on the lookout of different microbe strains that we can find. So lots of this kind of upstream discovery kind of process. Doing the sequencing are getting cheaper and cheaper now, handling the bioinformatics, things like that. Then when you selected a target candidate, then it's about the protein chemistry. So I myself work on membrane proteins during my PhD. In this case, the R&D scientists will be focusing on soluble proteins. Again, looking at the biophysicals, biochemical characterization of a candidate or a range of candidates, and then really understanding their behaviors, their optimal pH range, temperature range, their substrate specificity, and things like that. Then, let's say, very often we find a good candidate for a new product for, let's say, household care, a new protease, for instance. In household care, we typically wash our clothes at ambient temperature. Right? We don't really heat up. The penetration of the front loaders washing machine that you can increase your temperature is still very minimum in Southeast Asia. So the enzyme has to perform optimally at lower temperature compared to the enzyme used in Europe, whereby they typically heat up to 40, 50 degrees, or in the States, and long wash cycle. And in our regions, the climate can be quite challenging, so the warehouse can be quite hot. And then when you have logistics, you have to deliver the product to different customer or end user. They also can sustain high heat. Or even if you think about export market, to export to Middle East, which is also very hot. So then protein engineering comes into place to really, try to look into mutating different amino acids to find the optimum of structure whereby the function is still there, but then you have increased stabilization. And what Novozymes is investing is being able to do it in a high throughput manner than to do, I've done a few site-directed mutagenesis during my PhD, but in this case, we are doing it in a high throughput manner using robotics in the lab or different cutting edge tools in the lab to be able to then optimize that candidate. Then with all this process, you can imagine, right, there's a lot of data being generated, with the screening, with the assays, protein engineering, and also downstream processes whereby when we develop application, there are also a lot of data generated. So I routinely do wash tests in small scales or in a washing machine, and those will generate data, numbers, and things like that we need to capture more effectively in our digital pipeline so that we can then enable data science phase of discovery, artificial intelligence, machine learning, things like that. So we have a group of data scientists also looking at how we can better capture, store, make full use of all this data generated in our different labs or processes to drive future discovery of enzyme in different applications. This slide sort of illustrates a few trends, and then what we see from these trends, the opportunity that we see for enzymes or for Novozymes. So I mentioned just now the increased access to data. Right, data now, the different ways our social media is tracking our preferences, things like that. Automation. using robotics, or how can we then optimize certain processes, like in the production, like in car industry, how do we automate so that the human power is more in the design of the experiment, in the thinking, and is reduce the kind of labor cost to enable more high throughput ways of screening. And also the regulatory sort of environment is also getting more and more complex. Say if Novozymes can develop product quicker, we still also have to react to what is the registration of a particular product in a particular market, say in the food industry, which is a bit more stringent than household care, so that then the product can still be deployed effectively in the market, and checking all the things that require to be registered. And with all these sort trends, Novozymes are putting into effort into designing the way that how the whole company is putting the effort into making sure there's enough resources dedicated to address these trends, so that we can lead to opportunities. The first one I'm very interested is the tailored innovation, right? In the pharmaceutical industry, personalized medicine is a new trend. There's lots of PhD program looking into personalized medicine, looking at your gene sequences. Maybe you can eventually get a personalized medicine one day. In household care, unfortunately, I won't say that we have personalized solution yet. So each of us will still get the detergent that we get from the shopping mall or the shops that is nearby your house. But we do have tailored enzyme cocktails for a particular region. So we are in Southeast Asia. The climate is different from Europe. Our eating habit, our day-to-day life and our traffics and things like that are different. So then that requires tailor-made cocktail for different regions, and also sometimes tailor-made for different countries itself within Southeast Asia. Malaysia and Indonesia, our cuisines are maybe more similar. Bit oily, maybe we like the fried food, we like the spicy food, but in Vietnam will be very different, the challenges they face in their food habit. And with thinking about that, with the difference of data sciences pipeline, automation and screening, we hope then we can leverage across all these different sort of tools that we build in our lab to have better impact and also have better learning across the industry. So eventually we can then better define the space of enzyme within the industry. Sustainability, I'm glad that just now the person mentioned about sustainability. It's a buzzword, right? A lot of times when we talk to our friends and family, it's more about the environment, so being green to environment, sometimes vegan lifestyle and things. But sustainability is something that is really in the DNA of Novozymes. And I'm very pleased to say, if you check our financial report, Novozymes report last year, you can see that we specifically address how we address this UN Sustainable Development Goals in a few pages. And Novozymes is actually one of the first company to really embrace these goals when it's being proposed by the UN. So this one is really in our DNA. If we look further, sustainability is really a 360 demand, right? If you look into a definition and dictionary, it's able to continue for a period of time, right? Being sustainable, being maintained at a certain rate. Or if you look at sustainable development, is to able to meet the needs of the present without compromising the ability of future generations to meet their own needs. So now we are in a very special sort of situation where COVID is hitting. So whatever choices we make today has to be sustainable, do not jeopardize our future generations to meet their own needs. And it's not just about environment. There's also social and economic context to it and requires government action. For instance, climate changes, looking at how can we reduce the carbon dioxide emission. Corporate involvement like Novozymes, whereby the corporate strategy incorporates sustainability as one of the goals and then cascaded to our different KPIs of different employees in the company. And also that there has to be consumer demand. So for now, different region has different consumer demands, but I wouldn't say anyone would say, I do not want a product that can be beneficial to the environment and then also to benefit myself, doesn't jeopardize any economic opportunities, and there's no negative social context to it. And you can see this is definitely something that will be growing over time globally, and it will be picking up in our regions. Which brings me to this vision of household care and Novozymes. We aim towards 100% biological detergent. I'll further define this in subsequent slides. This is a vision that we recently set up just over a couple of years ago. And it's something that we are putting into effort to define this space. And what we're asking ourselves is to reimagine, rethink the way we clean in our daily life. In Enzymes, we believe that enzymes can contribute more to a detergent now than just having 1 or 2 enzymes or base enzymes in detergent to target certain stains. And enzymes are very weight effective chemicals, biochemicals, should I say. So very minimal amount of enzyme, so less than 1%, for instance, can bring about good cleaning benefit, which I hope I can show some examples later, compared to other ingredients that is also used in a detergent, which is definitely more than 1%. And hence, if the market is ready, say in Thailand, for instance, the detergents, they are typically very small in packaging, very compact in size. So their detergents are not filled with a lot of water in the liquid sense or fillers in the powder sense. It's basically filled up with the essential components and just reduce the packaging. So it doesn't require a lot of space on your shelf. And one thing nice is reduce the packaging, reduce plastic. So with biological detergents, we are not saying that we need to make 100% enzymatic detergent, so it's just water and enzyme. There are still the needs for other ingredients, say surfactants, builders, polymers in a very good detergent. But we think other biotech animal raw materials should also emerge, either from Novozymes or from other companies. Say biosurfactants is also a trend now, but it's still not cost effective enough to be really formulated for mass scale users, say the rhamnolipids or phospholipids. What about other types of biopolymers, for instance? Now the polymers are not derived from fermentation, right? So that innovation effort should focus on not just the core cleaning or the stain removal, but also how we can also work on whiteness, giving good whiteness benefit, deep cleaning beyond just food stains, for instance. There's something to be defined, and also reduce malodor, which is also a common laundry issue or challenge. And with this biological detergent vision, we hope we aim to provide great cleaning experience at a very competitive cost. So we don't believe that the consumer or our customer should pay a big price to make their product more sustainable, more green. And at the end of the day, it has to be better for us and also for the planet. So if you look into this slide, I'll further illustrate this biological detergent vision and the initial proof of concept. If you look at a typical cost of detergent, the raw materials will cost up to say 60% in general. Then you have your production and logistics and things like that. If you look at the typical common formats of detergents in our region, so in Malaysia, Indonesia, our powders are still dominant. We still like our powders, but then liquid detergents trend is on the rise. So that more and more consumers nowadays are looking into the convenience of liquid detergent and also certain benefits that liquid detergents can offer in contrast to powder detergents. But if you look at these two different formats of detergents, laundry detergents, 45-50% cost comes from this surfactant, this surface active agent that will really eats up the cost of the raw materials. And that is something that is being traditionally formulated and something recognized as something essential for detergent to have surfactants or mainly containing surfactants. But let's rethink cleaning again. Do we really need that much of surfactants? Can other biotech ingredients contribute more to a detergent formulation? And what are the challenges of having a lot of surfactants in a detergent? So if you look at this chart here, it looks a bit like a share price, but it's ready to show you the volatility of surfactant prices over a 10+ year period of time, right? In contrast to enzyme. So a y-axis show you the price index. Basically, this AEO, so this non-ionic surfactant derived from petrochemical. LAB, which is linear alkyl benzene, which is also a precursor to a very common anionic surfactant, LABSA, which is linear alkyl benzene sulfonic acid. They need to rely on petrochemicals, and then we know the prices of petrochemicals or fossil fuels, they are volatile in the market. In comparison to enzyme, whereby it's produced by fermentation. Prices do increase over time because of inflation. That's not something we can really control, but what we can control is there's a steady increase. So a detergent manufacturer can be better prepared to embrace this volatility whereby the bulk of the formulation depends on surfactant that is petrochemical derived. What about supply chain? There's lots of, global weather changes and things like that, and that can interrupt the supply of either a precursor to a surfactant or even the supply of the petrochemical surfactant itself. And with COVID-19 also, we see there's a shortage in containers and also disrupt the supply chains. So when your formula is predominantly based on surfactant, that can put a lot of pressure to the formulation scientists in the company. Whereas if you look into enzyme, which is still not effectively used, I would say, it's being used, but maybe we can expand the usage and enzyme further. Even at the worst week, right, Novozymes globally is still able to supply our enzyme to our customer, and there's less than 3% of a shipping delay. So in terms of sustainability, I would say the supply and then also the source of the enzyme is more sustainable compared to other chemicals used in the detergent formulation. And with that, I hope you somehow at least buys into the idea there's now a great opportunity to think about if the market is changing with enzyme being somehow maybe underutilized to a certain extent, or biotech ingredient being somehow underutilized. Let's rethink our detergent formulation to aim towards this vision of 100% biological detergent. And with that, then we can at least address to the growing needs of a consumer to want their product to be more natural, but still be powerful, better for themselves, better for the environment. And with COVID-19, we want a sense of safety, right? And in certain regions, then they are opposing to the artificial kind of ingredients and prefer a bit more natural derived. And we believe this trend will slowly seep into our market for consumer adoption. And that's why we set ourselves as a challenge. How can we decrease the use of petrochemical-based materials, say for instance surfactants, and knowing that their price is volatile, and address the needs of the increasing consumer demands on sustainability, but being able to deliver the excellent performance of detergents that the consumer are asking for at similar or even lower in cost. So it's quite a big sort of challenge, problem statement that we set ourselves, and we are starting to put in efforts in research and application to be able to solidify this concept so that eventually this is no longer a vision but a reality in the cleaning industry. So enzymes is different from some surfactants or from polymers. They are biocatalysts. They keep working. So from the moment you start clicking, turn on your washing machine, then your water start loading, you put your fabrics in, and then maybe you dose your detergent in, the enzyme will start working on the different stains they see. And then being a biocatalyst, there's thousands and thousands of reactions being generated over the period of the lifetime of the enzyme. Eventually it will denature, and that's normally when you flush your washing solution out of your washing machine and you start rinsing your clothes. Surfactants, you need a certain threshold of surfactants for myself, and then they will solubilize or emulsify the soil, but then they are tied out in that sense. Polymer ingredients, in all cases also being tied up, the particulates in the wash solutions, or they are being used to coat the fabric, and in that case it's not so much, or you can't really recycle them and to get to multiple reaction in comparison to enzyme. And that's the main reason enzymes are very weight effective. So minimum dose can give you a good cleaning performance. And with so many washing tests done over the years, we know that without enzyme, yes, say for instance, this blue bubble is a surfactant myself, goes on and saw maybe a curry oil onto a piece of fabric, right? And you can solubilize, you can, bring away, lift up the stains, fairly effectively. That's been routinely done. But with enzyme, let's say the detergent is formulated with say lipase and protease, that also works on the component in this say curry, that you dip your roti in this morning. Then it starts chewing up or digesting or hydrolyzing, the substrate, the specific substrate on the piece of fabric, and together work in synergistically with the surfactants and other ingredients. It will remove the stains better. This is a cartoon illustration, but it can be effectively demonstrated in washing tests that we have seen numerous times in our lab. And that's why we believe that we are able to demonstrate that with the use of right enzymes, the right enzyme cocktails, we can optimize and if not replace certain amounts of petrochemical derived surfactants to achieve similar or even boost the wash performance. So over the years, Novozymes has developed many classes of enzymes, and in this case, I'm showing you the classes of enzymes typically used in the household care industry. And fun fact, Novozymes was the first one in the 60s to sell the first protease into detergent industry. And then you have multiple new classes coming out. So we have the protease, amylase, lipase, peptide lipase, manganese, and also we have two types of cellulase. They work on cellulose, so cotton fibers that gives you more towards the fabric care kind of benefit. So it chews up tiny fibrils on the piece of fabric, and it can gives you the sort of prevent the dirt from depositing when you have these tiny fibrils. And what we see is, and also Dr. Pason mentioned just now, is using cocktails of enzyme, whereby you are using an army of enzyme in the right dosage, in the right way to tackle complex stains that we typically see in our daily cuisine and things like that. So it's not just on top of each other, but they work synergistically together with the other ingredients to really effectively remove your laundry challenge. To show you an example here, how we say that enzymes can effectively reduce the use of surfactant to a certain extent, but still boost the performance. I'm going to show you a test done here. So in this case, we have shortlisted 35 different types of stains, and those stains are being sourced from these different companies. They are institutes that supplies stains that all of us can purchase. So the detergent manufacturers like P&G, Unilever, the Lion, the Cow, the Wings, they can purchase these stain swatches for them to test. The beauty of these stain swatches are they are produced in a large quantity, so you can get the supply in a sustainable manner in a sense. They also quality control process to it so that you know that each batch of stains are quality control, so you can then compare result to a certain extent. And there's a wide variety, and it's in this industry, if we use the same stains, it's easy to talk about the technical jargons and then come to the same conclusion easily compared to, let's say, I want to test out the detergent on tomato ketchup, and we have different tomato ketchup in our kitchen. And it's due to resource constraint, you can make a lot of tomato ketchup to test a day. But using these things, then there's the feasibility of doing that. Give an example here. So you can see here, I just shortlisted two different swatches that is also listed in this stain set. So you have this rice starch, which is colored in this nice orangey kind of color. It's called CS28. This is a purified form of rice starch. So we require very technical, so it's only single substrate. But you can imagine that will measure the effectiveness of an amylase very readily. And with this, what we typically do is measure how much color is being removed from this substrate. So if you imagine the underlying fabric will be about white color, and then if you can remove 100% of this starch, then eventually it goes to white color. But that's not how these type of swatches are being developed. They are meant to give us a large testing window. So most of the time you see only some amount of color are being reduced in a consistent manner, and that's why we can compare a lot of different detergent formulations with different types of ingredients, different amounts of the same enzyme or different types of cocktails to come to a decision which formula is the best suited for this launch. And this is a purified component just on starch. What about chocolate ice cream? So in this case, it's more complex. So you have the protein component from chocolate, from milk. You also have the the gum that gives you the nice mouth texture when you eat ice cream, especially on a hot day. So in this case, that this wouldn't be just used to monitor a performance of amylase. This could be used to monitor the performance of a overall detergent with multiple enzymes in the formulation. Right. So one of the results that I want to show you now is about performance. I hope with talking over this slide, you can see that there's this potential of increasing the performance with the use of more enzymes, different classes of enzymes, and at the same time reduce the use of surfactant. First thing first, y-axis, you might not be familiar with these ways of plotting. So the y-axis shows you this total remission. Remission is the term we sort of combine to encapsulate the reflectance, because we are measuring fabrics. We are using spectrophotometers like this to measure how much light is reflected on a piece of fabric, because that is basically what we see. If we look at our clothes, how dirty they are, we are basically seeing the light reflected from that piece of clothes. So this is the key instrument that we use for stain removal or whiteness in the industry. And in cases, quite often that detergents are formulated with optical brightener. So it's not just reflecting, some lights are emitted out. So then we combine this as remission. And why we measure at 460 nanometer is strains back to the paper industry, whereby this is this thing called whiteness index. So they use this 459 nanometers or 460 to measure how white is a piece of paper. And in this case, you eventually want to get whiter clothes if the clothes is white, so it's very relevant. So we stick to this measurement at 460, and basically just sum up the different values you get from the 35 stains to this big number. And if you look at a starting formula, in this case 100% surfactant, that means there's no reduction in surfactant, and in this formula there's about 18% of surfactant in this, which is very common, maybe on the higher side in our region. It gives you this number. If you reduce the amount of surfactant by 30%, so dropping from 18 to about 12.6, 13%, then you will expect to see a decrease in performance or stain removal, as you can see. Because surfactants, again, is commonly used, is effective to remove stain paired with other ingredients. So then you will expect that decrease in their performance. However, if you look at when you decrease the amount of surfactant by 30% in general, then you start putting in enzyme blends. So besides your protease, now you put in amylase, for instance, a lipase, for instance, manganese or cellulase. They optimize this cocktail to around 0.48. So there's multiple rounds of study to come to these dosages. Then you start doing the same test again. You see that then there's a 5% of stain removal boosting from this, and we can repeat this in a lot of different ways by changing the amount of stains, the different types of stains, different formulation, different classes of enzyme that is tailored to that particular country or region. So we can do all this many ways, but we do have a lot of data to substantiate. Today, I'm just showing you one of these examples here. What about whiteness? So for whiteness studies, we use something called whiteness tracers, so that basically just different types of white fabrics here, different cotton or polyester or blends. Again, we measure the remission again of these 5 traces at 460. And because we need to add something to make the white fabric dirty, right? So in this case, we use liquid carbon black to mimic the kind of pollutants or dirt, the kind of outdoor pollutants that you typically see. So we add into the washing machine, and you don't get your clothes, white clothes being dirty in the first wash. Typically it's over time. So in this case, we have done multiple washes, as an example, and then again compare how white the fabrics, these traces still are across that, and what are the optimum formulation that can give you that the superior whiteness benefit. So show you some examples here. So if you again take the analogy of reducing the use of petrochemical derived surfactants, say by 15%, 30%, or even more drastically, if your starting surfactant is very high, then you can maybe reduce a bit more, up to 55. You see that actually the drop in whiteness performance is not as drastic as you imagine compared to stain removal, right? But then there's still drop, and that is something not surprising. But then when you start pairing, increasing the amount of enzymes or different classes of enzyme in the cocktail from 0.2% of protease, then you start saying, okay, let's take 70% for instance, and you put again the 0.48, then you already outperform your original whiteness index that you can get. And even if you reduce the surfactant by a large percentage, then you still see the boost in performance, but then it makes sense that you probably need a bit more enzyme in the blend, or maybe you stick with 50% of the surfactant change. So these are the things that we work on a sort of daily basis with our customer or in our lab to be able to illustrate the benefit of cleaning benefit of enzyme cocktails in the different formulations. So the application scientists would know how to formulate the different detergent products and be able to suggest beyond just talking about enzymes, and is to work around what do we get the optimum formula. And this is quite a commercial slide, but I hope this gives you a perspective that when we say that we don't believe the consumer should pay more for more.
I'm sorry. I'm sorry to disturb you, Dr. Alvin. We have 5 minutes to end your presentation. I'm sorry.
I can almost there already. Almost there already.
Okay, thank you.
Cool. So very quick. So 1 minute for this one. Just to give you an illustration. So we have done calculation to show that even when you increase the amount of enzyme, but with the reduction of surfactant, because the cost of surfactant is quite volatile, right, and can be quite costly, depends on then you can still get this 5% more or less cost reduction. So we believe that we are not just being arrogant and say that you don't have to pay more for that. There's a basis to that. And that's something we work also very closely with our customer. Let me summarize this presentation with touching on the environment. We also believe that by reducing these petrol-based chemicals or other ingredients not biotech ingredient, we can also address this UN Sustainable Development Goal. And I'll show you some example here. As a summary, we have global sustainability team that do life cycle assessment, calculation, and things like that. So ballpark figure is we believe that based on our initial calculation, and we are still doing that, reducing these chemicals like surfactant effectively reduce 10 times carbon footprint. Or in a sense whereby eventually our washing solution will have to reach our water sources, even though it's being treated, less amount of water, 10 times less, needs to then dilute these chemicals so that it's not toxic to the aquatic life. 3 slides and I'll end. This is an example whereby if you look at, again, the same example, if you reduce 30% of surfactant, this is the amount of carbon dioxide you can reduce compared a gram per wash, right? And doesn't seem a lot, but this is one wash itself. And if you add in the extra amount of enzyme, it doesn't give you a lot of carbon dioxide because of the nature of enzyme being, the way it's being produced, the nature of enzyme itself. But then if you extrapolate, right, so if you take this one, ideally, if you reduce 30% of surfactant across the world, you can effectively reduce 1.2 million tons of chemicals out, and then that can equate to 3 Empire States building in the US. So it can be quite astronomical. How about carbon dioxide emission? Think about a congested city like Jakarta, like Kuala Lumpur. If you reduce 30% of surfactant, then you effectively reduce about, say, 11 grams per wash. And then the amount enzyme added is still 10 times less. And in this case, that you are effective, if you do the calculation, extrapolate globally, you are taking out millions of cars off the road. And that can be quite significant because we don't only cover just the usage, the production, the surfactants and things like that. Last but not least, this is also a fairly new concept to me. It's called critical dilution volume. Basically, it's the amount of water needed to dilute the detergent ingredients so that it's not toxic to water living species. Huge number here, but same principle here. If you take out this, you can reduce this amount of water needed to really dilute the wastewater so that it's no longer toxic. And then the use of enzyme, it doesn't really cost so much because they're really biodegradable, they are from nature. And that is also a huge number to ponder on. So I hope with my presentation, you are maybe a bit convinced or at least start thinking about this biological detergent, in contrast to the conventional fully loaded with surfactants, can also give you good cleaning experience. Price also we can manage so that the consumer don't have to be paying extra cost or high cost to it. And eventually this is better for our own use and better for our planet. And I hope that you can rethink how we clean together. And with that, I thank you for your attention, and I'm happy to take any questions.
Thank you very much, Dr. Alvin. I really enjoy your presentation, and hopefully everyone here also can get the knowledge that's shared by Dr. Alvin. And now is the Q&A session. Now Dr. Alvin will answer some questions that were sent to us during the presentation. And as a reminder, you can still submit the question through the chat panel. And we have around 50 minutes in this Q&A session. Okay. The first question is, can share the question, please? Okay. So the first question is from Dr. Nanik Rahmani. The question is, how long does it take to develop a new enzyme until it's ready for application? And are all Novozyme products is a recombinant enzyme?
Very good question. So if you recall one of the slides, typically a new product development, when there's a need, there's a business need, also has to justify the business potential. It takes about 5 years from the initial screening, looking into our database, say we want a new protease for household care, and down to at the end ready to deploy to my hand so that I can then present to the potential customer together with my sales and marketing colleague. Take about 5 years or so. But I think the vision of Novozyme is to shorten it down to maybe 3 years. And that has to be really guided in the screening stage, whereby we need more data scientists to help inform us. And also we need to minimize the amount of wash tests. That means we need modeling to help then guide what are the critical wash tests that needs to be conducted to give us some forms of understanding whereby we know that this product is definitely better. Are all enzymes recombinant? There's certain extent enzymes are recombinant. Some of them are wild type. But wild type enzymes, eventually we could also protein engineer it so that it's more stable. So I can't answer on top of my head, but I would say it's a mix of them. And certain industry recombinant enzymes are not allowed, then in that case will be wild type.
Okay, thank you very much, Dr. Alvin. So it's take around 3 to 5 years.
Yeah.
Okay.
I would say 5 years or so, but I think we can aim for 3 years maybe eventually.
Okay, thank you very much. We move to the second question. To Dr. Alvin, you have already explained about the enzyme discovery. Would you please explain about the enzyme production, about the scale-up production of the enzyme?
Okay, I'll try my best because I'm not working in the production, but I've been to Novozyme's production. Basically, once you have the right candidate, you need to select the right host, the right, either a bacteria or fungi to produce your enzyme. Then you optimize your fermentation process, and that is done from pilot plant, and then you scale up to our big fermenters in the production. Then after that, then you need to purify the enzyme away from the host, right, and away from other the food source from that. So then we have purification process in place. I can't tell you exactly what are the types of membrane used, but if you are interested, I can check with my chemical engineers in the plant. Then after that, then depends on what is the end application of the product. So detergents are being used in both powder detergent and also liquid detergent. So in powder detergent, you need to make the enzyme into granules. So then we need to make them into granular form. So then there's coating of different salts and different matrices, so that then they are like biji, tiny balls of granules to be then dosed into your powder detergent together with your surfactants and this. Liquid detergent more straightforward. Then you take your purified enzyme, then you put in some polyols, some preservative, for instance, so that then it comes into bottle. Then the customer can then dose into their mixing tank to make the final detergents. So that is the case whereby is in a snapshot how enzymes are being produced in our different plants across the world. Then they are being packed into different pack size, depends on needs, and then ship across. Most of the times through ship. In urgent cases, then we will air freight them to our customer. And the scale of production, you are talking about tons of enzymes being produced per batch. Tons, yeah.
Okay, thank you. If I'm also interested with this question. So usually what host is used for the production of enzyme in Novozyme?
What's the, sorry again, what's the workhorse mean? What strain?
I mean host cell production.
This is, I would like to answer your question spot on, but again, this is something that I also won't have exact knowledge because it's quite covered by IP. So being a biotech companies, this is something that will cover in our patent, and we have a range of host strains that is optimized in a particular factory. So I can tell at least even if the same product being used in Indonesia, for instance, or in Denmark, they could come from different hosts because that fermentation has been optimized for that. But I would say bacillus will be a common species being used, and but we also have fungal strain aspergillus. And then I can't tell you the down to the exact level or what is the changes to that. There are scientists looking into optimizing fermentation on a day-to-day basis. And then same product also can be produced using different host strains. But at the end of the day, what we are looking at is that molecule, right? The molecule has to have that 3D structure, the amino acid. So then it doesn't really, it's not so much derived from what host it comes from. It's more like a production house to get to the enzyme at the end.
Okay.
Yeah.
Thank you very much. I hope it's helped. We move to the third question from Mr. Untung from Pak Untung M from Rajawali Company. So the question is, if my company intend to build a 10 KLPD2G ethanol from sugarcane bagasse, could you supply the required enzyme at what cost?
It sounds like I'm going to see a customer. Thank you so much for this question. I'll say please drop me an email if you can't see in the beginning. Please write to me. I will connect you to my colleague in this industry because I'm from household care, right? But we do have colleagues looking into this bioethanol. I assume 2G is bioethanol, and then in this case, my colleague can even help maybe optimizing the process and things. Novozyme is also an industry that Novozyme takes care of in this industry. So please drop me an email. You can check with the organizer, and then I will pass you to the relevant colleague for sampling, costs, and things like that.
Okay, thank you very much. Question related with this will can send email directly to you, right?
Yes, that, and I'll pass on to my relevant another technical colleague in charge of this industry.
Okay, nice. Thank you very much.
No worries.
Okay, we move to next question from Pak Harto Widodo. So an enzyme is a substance that accelerates the reaction without reacting in a biological reaction. When is the enzyme damaged during the reaction cycle, and what causes the enzyme to be exhausted? And what can we do to increase enzyme durability?
Oh, I really like your question. This is spot on. I get this question also once in a while. So let's go back to the example of protease again, right? Proteases are acting on protein. So and for household care, proteases are typically quite broad range. So it's not just targeting on your milk. It works on your blood, on grass, on meat, for instance. So enzymes, for enzymes to perform, they need to have their 3D structure folded, right? At least in solution, so that the active sites are not blocked or they are not being hydrolyzed by other proteases. So one of the challenges actually, when you have a group, thousands and thousands of millions of proteases in a formula, they can start attacking each other, at least in the liquid detergent context, because they are not protected like in a granule, and there's water. So most of the classes of enzymes in household care, they are hydrolyzed. So water is present in the active site, then the catalytic reaction will occur. So in these cases, during the washing cycle itself, because the sheer amount of other substrates are more than the protein itself, the proteases typically will go and work on other protein stains like your blood, like your milk. But over time, a protease will also react on each other, then they themselves are destroyed. Or the other ingredient in the wash can also distort the 3D conformation And because you don't have other sort of chaperones to help refold them, then they are also lost. But good thing is they are recycled. So those active ones can still work on. Typically what we see is if you simulate a wash sticker within, say, after 1 hour in that washing liquid, and then we remeasure the enzyme activity, you see some losses already, or temperature. So in terms of shipping, like I mentioned, the heat itself, thermal degradation, right? Because you are increasing kinetic energy, it also will then destroy the enzyme over time. How do we then increase the enzyme durability, or what we say is stability? Coating in the powder form, that would definitely increase the durability against the surfactants, the bleach or other ingredients, or humidity or temperature. In liquid, for enzymes that is used in the liquid detergent, what we do is we use peptide inhibitor. So Novozymes also have a patented technology whereby there's few chains of amino acids that's very sort of special design. I can't tell you the exact chemistry, but then it blocks active site. Then together with, just now I mentioned polyols, like monopropylene glycol and things, or some other salts like sodium formate, calcium chloride, because our enzymes also have calcium binding site in the 3D structure. All this comes together into the final formulation so that you keep the enzyme stable until it's being used in your bucket of water, if you hand wash, or into your washing machine. And then once you finish your main wash, then even the enzyme is degraded, then it's okay, because then they have done their job. But you don't want the enzyme to be degraded on the shelf, in the packaging, or during the shipment itself.
Okay, thank you, Dr. Alvins. I think it's very clear answer. And then we move to the next question. Is the question from Pak Rizky Alfarizi. So the question is, I'm very interested about Novozymes development in detergent product, and I think it will be good for our environment. And the questions are, do the composition of chemical material in detergent not effect for Novozymes work? Do Novozymes are safe when contact directly with our skins?
Okay, also two quite common questions that I face also. So like I say, so being a formulation or applications of a scientist specialist in here, we do know the ingredients used in detergent formulation, and we quite routinely also subscribe to databases whereby what are the common ingredients, or do our formulation analysis through third parties. So we do know the typical surfactants, polymers, things are being used in the industry, and certain ingredients will be more detrimental to enzyme. For instance, if you use a antibacterial agent, or things like ethanol, for instance, if you use high percentage of ethanol, then you can imagine it's meant to kill things. It will also destroy a biochemical like enzyme, or some preservatives. Formaldehyde, for instance, will also destroy the enzyme. So those kind of knowledge we do have, and typically what we have conversation with our customer is we will give them some forms of formulation guideline for the liquid product, because it can be more challenging, and we will work with them to run accelerated stability tests so that we can do 1 to 2 months high temperature to try to simulate 1 to 2 years of shelf life. Depends on the company's of success criteria, and it's iterative. So constantly we are doing all this, and then we will analyze at the end of the storage condition, what is the activity. So we do a biochemical assay on what is the performance. So again, I'll take some swatches like the CS20 I show and check and show to the customer, hey, the amylase are still working, right? Even though maybe you see a drop in the activity, because again, enzymes, they are recycled. They can still function within that given wash time, which is pretty much 15-20 minutes, more or less in a main washing machine. So we do have the expertise to work with, and that's why we see that this is also a value of Novozymes bringing into this cleaning space. Not just about our enzyme being a good product, but also how to apply it optimally in the industry to go towards biological detergent. Second question is about safety concern. This is something also we've been asked, and in terms of production, there's very sort of like guided way to handle enzyme in the production. So of course with PPE and things like that. So as long as the company conforms to, you don't just touch the enzyme bare hands, but it won't kill you, but maybe you might get sensitization, like other allergens that we face. Some people are sensitized to pollens and things like that. But there are already this safety documents being generalized. This thing called HERA document, risk assessment, and they have shown that this is done in the UK many years ago, decades ago, that the dosages of enzyme, so less than 1%, I've shown you an example, is too little, even if you have direct contact of a detergent product, to really cause any sensitization for the general public, unless you are really sensitized to this enzyme, right? And before launching any product, you have to be rest assured, our safety department will have analyzed toxicology kind of tests to make sure that in the typical usage of an enzyme in a detergent product in this region, things are covered. So you shouldn't face this kind of complaint from a customer. And you might worry that actually, if you worry about the consult enzyme on the skin, I will actually question that some other surfactants are even more harsher to your skin, or pH of a detergent can be harsher. So I think enzyme in itself, we know what we can help, but it shouldn't be the main concern of about that. The other chemicals can be a bit more challenging to handle.
Okay, thank you very much, Dr. Alvin, for your clear answer. I think it's give reassurance to us. And then I think there's one last question from me. It's okay?
Yes, no problem.
Oh, thank you. So my question is, previously you explained about the application of mutated enzyme in the Novozyme, right?
Mm-hmm.
So is there any specific regulation about the application of the mutated enzyme? Because maybe some concern about the use of mutated enzyme, or we can directly apply it?
It will depend on that. It's a complex question. That's why we also have a regulatory team sitting in Kuala Lumpur itself that caters for the Asia Pacific region. So our regulatory colleagues will constantly look out new guidelines. And sometimes, when the country develops, like Malaysia and Indonesia, more things will be put in place. But we make sure that the new law put in place has been properly formulated with an expert opinion. And in some countries, by default, GMO are not allowed, things like that. Then we do have products, and that's why we need to know the business case. If we are tackling, say, a food industry whereby GMO is a no for the time being, and we don't foresee that the law will change in the next 5 to 10 years, we won't go into a 5 years development pipeline to go and engineer that. But in household care, at least within Southeast Asia, it's not so much regulated in that context. Maybe then there's some, like Indonesia, I understand there's some amounts of a minimum amount of surfactant needs to be there. That's why people are overly loading on surfactants. That's the conversation I heard. So that then they at least reach the minimum, because then they need to make sure you're not just selling water to your consumer. So there's the government sort of protecting the end user, right? But in that case, the enzyme is not factored in that. So in this case, we have a bit more freedom to operate, and it's not like we autopilot going to protein engineering. Protein engineering is there, maybe they find a way to stabilize, and if the molecule by default, wild type, doesn't need any specific stabilization, then why do that?
Oh, yes.
Yes, so that it really needs to fit. But I think, as we all are scientists, right? We know that, and CRISPR is coming out, and one day will, if you use it in the right manner, will really benefit the biotechnology and environment, medical sectors or industry. It's more having education, the younger generation or the government sector about what should be done and what can be done. As long as what I'm seeing, the enzyme should really focus on the substrate on a piece of clothes, so that once you wash your clothes, you dry your clothes, you can be assured that, oh, this is a nice clothes I can wear, your baju and things like that. That should be fine. And I don't think that should be too much of a concern.
Yes, I agree with you. Because I have great interest in protein engineering also, so I would like to ask your advice about that. Thank you very much, Dr. Alvin. Now we end our Q&A session, and once again, thank you very much, Dr. Alvin, for coming and share your knowledge and give us some insightful advice. I hope it will benefit all of the participants and attendees here. So for the next session, I will give back to Mbak Aulia. Thank you very much.
Thank you.
Thank you very much. Apologize for the inconvenience matter. Thank you, Dr. Alvin Teo, for the informative presentations this morning. Also, thank you for Dr. Vina to lead the first session. Ladies and gentlemen, we will have another break for 5 minutes before having the second plenary sessions this afternoon. Please stay in the Zoom and come back again at 11:15. Thank you. Ladies and gentlemen, welcome back to the webinar. Since Dr. Alvin Theo and also Dr. Patra Pessen have other agenda, they will leave the room this afternoon. Thank you to Dr. Alvin and also Dr. Pessen for joining and also giving us insightful information for today's webinar. Hope you always in a good health and also stay safe. Distinguished guests, ladies and gentlemen, proceed to the next agenda. We will continue to second plenary sessions that will be conveyed by Head of Center of Excellence for Integrated Biorefinery, Research Center for Biotechnology LIPI. May I kindly invite our moderator, Dr. Vina, to lead the sessions. Dr. Vina, time is yours.
Thank you very much, Mbak Aulia. Hello, everyone. Thanks for staying in this webinar. In these sessions, I would like to welcome Dr. Eng Nani Rahmani. Hello, Dr. Nani.
Yeah. Hello.
Okay, thank you very much for coming today and share the knowledge with us in the next presentation. So before we begin the presentation, I would like to introduce the biography of Dr. Eng Nani Rahmani. So Dr. Nani now is the head of Center of Excellence for Integrated Biorefinery Research Center for Biotechnology LIPI. And she finished her PhD in the Chemical Science and Engineering in Kobe University, Japan, and her research interest is in biotechnology, bioprocess, biorefinery, molecular biology, fermentation, and enzymology. And if anyone interested in her publication, also can check in this slide. Okay, thank you very much. And now, without any further ado, time is yours, Dr. Nani.
Apakah sudah
Not yet.
jelas? Sudah kelihatan belum ya?
Presentasinya belum, Bu Nani. Oh, oke.
Sudah ya?
Sudah. Clear.
Oke. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang. Sebelumnya mohon izin saya presentasi dalam bahasa Indonesia. Jadi pada kesempatan yang baik ini, pada seminar pertama web series Enzyme, kami dari Pusat Penelitian Biorefinery akan mempresentasikan tema The Current Technology and Application of Enzyme in Food Industry. Outline secara garis besar presentasi saya yang pertama, sejarah gimana sih aplikasi enzim di industri pangan yang sudah berlangsung mungkin beberapa abad sebelumnya, kemudian perkembangan enzim di industri pangan untuk saat ini, kemudian bagaimana strategi untuk mendesain enzim yang khusus untuk aplikasi di industri. Kemudian pangsa pasar enzim secara global di dunia, kemudian aplikasi enzim untuk food industry itu seperti apa aja, dan selanjutnya proses produksi enzim untuk aplikasi khusus di food industry, dan perkembangan riset saat ini yang sedang kita lakukan, terutama untuk aplikasi di food. Sebagaimana kita ketahui bahwasanya aplikasi enzim di industri pangan ini sebetulnya sudah berabad sebelumnya ya, udah banyak dirintis, yaitu dari tahun 1830-an, di mana enzim yang dipakai dalam industri pangan itu baik berasal dari tanaman, mikroba maupun hewan. Yang udah kita kenal dengan baik yaitu renin, yang di mana ini banyak dipakai dalam industri keju, dairy industri. Nah, kemudian berkembang terus sampai tahun 1980 itu mulai masuk dengan pendekatan genetic engineering. Nah, kemudian untuk saat ini kita di dunia ini mengembangkan enzim untuk aplikasi di pangan itu apa sih? Yang pertama yaitu prosesnya biar lebih mudah, kemudian secara efisien menggunakan bahan bakunya, kemudian untuk kualitas produk juga secara konsisten terjamin, kemudian memperbaiki sustainability tadi sudah diterangkan juga oleh Dr. Tio, jadi ketersediaan keberlanjutan dari enzim ini untuk aplikasi yang kontinu di industri pangan. Nah, tapi untuk saat ini ada tambahan penekanan selain sustainability, yaitu tren ke arah kesehatan. Nah, perkembangan selanjutnya dengan enzim dengan genetic engineering, karena dengan genetic engineering ini akan banyak memperbaiki dari proses produksinya, kemudian cost efektif dan kita bisa mendesain enzim yang sesuai dengan kebutuhan untuk industri pangan. Jadi kita akan memasuki new era enzim teknologi di mana menggunakan perkembangan baik itu teknologi biologi modern maupun chemical. Nah, bagaimana strategi untuk mendesain enzim untuk aplikasi di industri? Jadi skema teknologinya bisa dilihat pada slide berikut. Jadi yang pertama adalah gimana kita menemukan enzim yang novel, yang baru, yang unik, yang dia spesifik untuk aplikasi tertentu. Nah, ini pendekatannya ada dengan metagenomik untuk enzim-enzim yang dia itu sulit didapatkan dengan culturable. Emang di dunia ini kan ada 99% mikroba yang tidak dapat di-culture. Jadi pendekatannya dengan metagenomik, kemudian dengan screening biasa. Nah, dari sini kita akan mendapatkan natural biocatalyst atau enzim yang spesifik sesuai dengan target yang akan kita inginkan. Nah, setelah kita mendapatkan enzim, nanti kita akan cari strukturnya, identifikasinya. Nah, kalau misalnya kita sudah mengetahui dengan homologi modeling, kemudian kalau yang tidak kita tahu, kita menggunakan struktur determination dengan NMR, X-ray crystallography. Nah, bagaimana selanjutnya untuk upscaling-nya? Biasanya untuk upscaling-nya yaitu pendekatannya dengan heterologous expression system. Jadi dengan enzim yang sudah kita screening tadi untuk bisa dia high productivity, high activity, dan lain-lain, itu kita cloning gennya, penyandinya, kemudian kita ekspresikan di host yang sesuai untuk aplikasi di industri. Kemudian pengembangan dengan menggunakan bioreaktor. Nah, terakhir selanjutnya ketika kita pengen memperbaiki dari misalnya suhunya mau biar dia lebih tahan suhu tinggi dan lain-lain atau spesifik substrat. Nah, itu ada beberapa teknologi sign direct mutagenesis, direct evolution, immobilization, teknik ini yang bisa kita kembangkan. Nah, kemudian strategi tadi jadi ada 3 proses yang bisa dilalui. Yang pertama adalah proses engineering-nya. Jadi di proses engineering-nya itu kita bisa mendesain bioreaktor yang maksudnya untuk optimize produksinya di skala bioreaktor. Kemudian optimize kondisinya, misalnya pH-nya, suhunya, aerasinya, RPM-nya di fermentornya, kemudian analisis teknoekonominya. Kemudian yang kedua, dengan menggunakan immobilized enzim. Nah, dengan menggunakan ini kita bisa memperbaiki stabilitasnya, bisa dipakai ulang, kemudian fungsionalnya juga. Kemudian kita juga bisa menghemat proses ketika proses pemisahan. Kemudian molekuler simulasi untuk mendesain biokatalis yang optimal. Nah, terakhir kita bisa menggunakan pendekatan dari rekayasa protein engineering. Nah, ini bisa meningkatkan biokatalitiknya, memperbaiki affinity substratnya, kemudian solven dan termostabilitas dari suatu enzim, kemudian juga bisa meningkatkan yield dari produksi enzim yang kita produksi. Nah, selanjutnya setelah kita mengetahui tadi sedikit histori terkait enzim di pangan, nah kita bisa melihat pasar global enzim di dunia itu seperti apa. Jadi kalau melihat jumlah enzim yang khusus diaplikasikan pada industri pangan, itu market size-nya pada tahun 2019 $1,9 juta, dan ini terus meningkat terus dan diprediksi tahun 2026 meningkat menjadi 3.000 juta dolar. Nah, peningkatannya itu annual growth rate-nya itu per tahun bisa mencapai 5-6%. Nah, ini sangat memberikan keuntungan ekonomi yang sangat besar untuk industri. Nah, dari total mungkin sekitar lebih dari 400 enzim yang berkembang di industri itu, 158 itu enzim yang diaplikasikan untuk pangan, yaitu meliputi 29% total dari enzim di marketnya. Nah, kenapa ini di pangan ini mungkin paling tinggi ya, meningkat terus kayak gitu tiap tahun? Itu karena ada beberapa faktor, yaitu terkait concern dengan safety dari lingkungan, urbanisasi, kemudian perkembangan dari research and development activity, kemudian perkembangan dari teknologi enzim sendiri yang meliputi rekayasa enzim, kemudian rekayasa enzim ini yang khusus memang kita bisa menghasilkan enzim yang spesifik untuk suatu kebutuhan di industri pangan tertentu. Kemudian juga beberapa keuntungan positif dari beberapa enzim yang aplikasinya sudah sangat banyak di industri pangan. Kemudian perkembangan dari inovasi teknologi sendiri, di mana dengan menggunakan enzim kita bisa mengurangi dari penggunaan chemical. Nah, kalau dilihat market share-nya ini aplikasi enzim di industri pangan, yaitu di pangan dan beverage ini yang paling tinggi, ini yang warna ungu tua, kemudian di deterjen ya tadi dijelaskan oleh Dr. Theo, kemudian di pakan, biofuel, tekstil, pulp and paper, nutraceutical, personal care, and cosmetic, kemudian water, dan yang lainnya. Nutraceutical ini termasuk salah satunya juga yang oligosakarida atau rare sugar kayak gitu. Nah, kemudian kalau dari market share-nya berdasarkan type-nya tadi di awal, jadi beberapa enzim kan macamnya banyak, ada karbohidrat, protease, lipase, dan lain-lainnya. Nah, dari market share-nya memang yang paling dominan dari tahun 2019 sampai sekarang ini, yang mendominasi di pangsa pasarnya adalah kelompok karbohidrase. Jadi dia enzim-enzim pendegradasi karbohidrat ini hampir lebih dari 50%. Di antaranya ada amylase, silanase, manganese, and pectinase. Nah, kemudian berdasarkan source-nya, jadi market share-nya dari sumber gimana enzim ini kita dapatkan. Jadi tadi dijelaskan kalau enzim itu bisa dari tanaman, dari hewan maupun dari mikroorganisme. Nah, hampir 90%, hampir 80% lebih ya, itu market share tertinggi memang enzim yang bersumber dari mikroorganisme. Nah, ini kenapa? Karena dengan menggunakan mikroorganisme kita bisa scale up produksi sampai beratus-ratus liter maupun ton dalam skala yang besar. Kemudian kita dapat melakukan modifikasi tadi ya dengan pendekatan molekuler maupun biokimia dengan sangat mudah. Kemudian kita juga bisa melakukan hiperproduksi, misalnya dengan untuk mendapatkan yang aktivitasnya tinggi dengan menggunakan overekspresi dari enzim yang kita temukan untuk di host yang sesuai kayak gitu. Kemudian environmentally friendly udah jelas ya, jadi ini mengurangi penggunaan chemical, kemudian menghemat energi, air, kemudian bahan baku juga, dan juga menghasilkan limbah yang lebih sedikit, kemudian lebih stabil dan aktif dibandingkan enzim yang dari tanaman maupun hewan. Selain itu, enzim dari mikroba ini kita bisa produksi secara efisien dengan beberapa teknik fermentasi, bisa dia solid state, submit fermentation, fermentasi semi cair atau padat. Nah, ini tergantung dari tujuan kebutuhan enzim yang akan kita gunakan. Nah, ini sangat menghemat cost baik dari waktu maupun dari space-nya gitu. Nah, kemudian apa sih potensial dari enzim yang diaplikasikan untuk di industri ini? Kenapa fungsinya tadi yang paling besar mendominasi? Sebenarnya fungsinya untuk apa? Jadi enzim yang digunakan di food industry ini merupakan kompleks ingredient. Jadi dia diaplikasikan terutama sangat besar itu tadi ya di bakery, dairy, kemudian untuk yang pengembukan daging, kemudian beverage minuman, juga sugar dan oligosakarida. Nah, ini fungsinya enzim itu untuk apa? Yang pertama sebagai dia kataliser, kemudian meningkatkan dari keragaman, varietas, kualitas dari selama proses produksi suatu produk makanan. Kemudian juga meningkatkan tekstur, flavor dari suatu produk makanan. Kemudian terkait untuk preservasinya, penyimpanannya, koagulasi, kemudian di daging tadi ya untuk membantu dia dalam keempukan daging kayak gitu. Nah, ini beberapa contoh aja enzim di beberapa industri pangan, salah satunya ini di bakery. Jadi di baking itu dalam proses pemanggangan untuk pembuatan roti, enzim itu ada 3 ya penggunaan enzim. Yang pertama dia memang udah alami ada di tepungnya, kemudian yang dia berasosiasi dengan mikroba. Contohnya mungkin contohnya di ragi yang digunakan kebanyakan untuk industri kue, kemudian yang kita tambahkan dari luar. Nah, ini yang kita tambahkan ke dalam adonan kue. Jadi aplikasi enzim di bakery ini bisa memodifikasi dari areologi adonan, kemudian juga retention gas, kemudian kelembutan dari proses pembuatan roti itu sendiri. Kalau di dalam pastry and biskuit, dia juga memodifikasi untuk reologi dari adonannya. Kemudian kalau dalam pembuatan cake itu dia memperhalus produknya smooth. Nah, di industri pangan ini enzim biasanya dipakai bisa individual atau sinergi. Contohnya di roti baking biasanya kombinasi misalnya antara amilase, silanase maupun dengan protease. Nah, kemudian kalau di dairy industri, nah ini juga banyak banget enzim yang digunakan di industri pengolahan produk perah itu dalam pembuatan keju. Salah satunya dalam pembuatan keju, terus es krim, yogurt, kemudian beberapa frozen dessert ya. Nah, ini beberapa enzim yang digunakan ada lipase, proteinase, kemudian laktase. Nah, di proteinase ini dan lipase dia di CHES misalnya dia akan memberikan flavor yang excellent, kemudian juga mengurangi kepahitan, level kepahitannya. Kemudian kalau di laktas dia misalnya untuk membuat susu yang no laktos untuk biasanya pasien atau yang dia alergi terhadap susu sapi diantaranya. Kemudian enzim di pengembukan daging. Jadi metode pengembukan daging biasanya ada beberapa ya, dia secara natural atau dengan stimulasi elektrik atau dengan mechanical atau dengan induksi tambahan dari enzim proteolitik. Nah, ini dalam fase tenderization ini adalah waktu yang tepat dalam menambahkan enzim protease di dalam proses pengembukan daging. Kemudian di industri minuman ini sangat banyak banget ya aplikasinya, baik itu di jus, pembuatan bir, wine. Nah, ini terutama enzim pektinase, selulase, sama tanase. Nah, sebelumnya penggunaan maksudnya tradisional yang tanpa enzim dengan enzim. Biasanya kalau dengan treatment enzim, dia akan meningkatkan dari yield-nya dari jus yang kita dapatkan, terus kejernihannya, kemudian total padatan yang terlarut di dalam jus, kayak gitu. Kemudian viskositasnya juga, kemudian memperbaiki dari pencairan dari pulp-nya. Nah, biasanya dalam proses pembuatan jus itu, kekeruhan itu karena adanya pektin, selulose, starch, protein, tanin, lignin yang ada dari buah yang kita jus. Nah, untuk menjernihkan itu perlu beberapa enzim yang biasa dikenal dengan kalau di komersialnya itu istilahnya macerating enzyme. Nah, ini ada beberapa yang biasa digunakan yaitu pektinase, selulase, dan tanase. Ini contohnya kalau sudah digunakan enzim kayak jus apel yang kita biasa beli ya, itu kan jernih banget kayak gitu nih, salah satu contoh aja. Nah, kemudian enzim untuk produksi oligosakarida. Jadi kebetulan di lab kami memang selama ini fokus produksi beberapa oligosakarida dari beberapa biomasa yang ada di Indonesia. Salah satunya galacto-oligosakarida. Nah, ini menggunakan beta-galactosidase. Malto-oligosakarida ini menggunakan enzim amilase. Kemudian mano-oligosakarida ini menggunakan enzim mananase. Nah, silo-oligosakarida menggunakan silanase. Kemudian inulin oligosakarida ini menggunakan indo-inulase, dan pekti-oligosakarida menggunakan pektinase. Kemudian silo-oligosakarida menggunakan selulase. Nah, ini contoh beberapa enzim yang memang paling mendominasi di industri pangan, diantaranya alpha amylase. Alpha amylase ini, amilase ini merupakan enzim pendegradasi yang dia itu menghidrolisis, mendegradasi polisakarida dari ikatan glikosidik yang ada di start itu. Banyak dimanfaatkan di baking industri, kemudian untuk memproduksi gula sirup di industri gula, dia produksi glukos sirup, high fructose corn sirup, maltose sirup, dan dia juga mengurangi viskositas dari sirup dari gula itu. Kemudian untuk penjernihan jus juga dipakai, untuk daya simpan dari jus juga dipakai amilase ini. Kemudian silanase ini juga mendominasi enzim di industri pangan. Aplikasinya sangat banyak. Saya kebetulan udah beberapa tahun ini riset silanase. Ketika waktu itu sempat ngecek beberapa jurnal, ternyata saya pikir silanase ini hanya dipakai untuk misalnya di industri deterjen, pulp paper, bioetanol, dan lain. Ternyata dia itu banyak banget untuk diaplikasikan di industri pangan dan di industri roti, baking, kemudian di pembuatan biskuit, kemudian untuk di industri kopi juga. Jadi banyak banget aplikasi dari silanase ini ternyata di industri pangan. Kemudian lipase. Nah, lipase ini nggak sebanyak silanase sama amilase, tapi dia banyak banget juga, lumayan banyak diaplikasikan di industri pangan, terutama untuk di dairy product, kemudian di butter, margarin, kemudian juga untuk masa simpan dari baking, produk-produk roti kayak gitu. Terus untuk di industri kelapa sawit, dia untuk misalnya ekstraksi CPO yang dia untuk menghasilkan asam lemak yang sehat kayak gitu. Nah, kemudian bagaimana sih proses produksi enzim untuk aplikasi di industri pangan? Jadi selama ini hampir kebanyakan untuk aplikasi di industri itu diproduksinya dengan submerged fermentation atau fermentasi yang cair. Nah, ini gambaran. Jadi kita menggunakan seperti ini bioreaktor. Biasanya kalau di industri itu karena dia mungkin ratusan liter atau sampai ribuan liter, jadi media itu udah langsung masuk di tangki fermentornya dan disterilisasi di situ. Terus baru nanti setelah dingin dia untuk diisi dengan dari seed culture-nya. Nah, kebetulan di biotek kita punya fermentor yang seperti ini, skala untuk 10 sampai 15 liter. Kita ada fermentor seperti ini. Nah, proses produksinya itu yang pertama ya proses fermentasinya, kemudian itu recovery enzimnya, purifikasi dan formulasi yang bagaimana untuk nanti aplikasi di industri. Nah, jadi 4 proses ini masing-masing berdiri sendiri, tapi secara integrasi untuk satu kesatuan untuk menghasilkan enzim yang siap untuk diaplikasikan di industri. Nah, proses fermentasi sendiri ada beberapa proses cara gitu. Seperti batch fermentation, ini biasanya semua media masuk semua langsung start fermentasi. Nah, ada juga yang feed batch, ini sama seperti batch tapi ada penambahan selain dengan beberapa penambahan media selama proses fermentasi, misalnya ada penambahan trust mineral atau sumber nitrogen dan lain-lain gitu selama proses fermentasi berlangsung. Nah, yang terakhir continuous fermentation. Jadi ini proses fermentasi yang nanti kayak recycle medium gitu, selnya dipanen, terus nanti setelah itu selnya itu bisa dipakai lagi dengan penambahan fresh mediumnya. Contohnya yeast. Nah, kemudian ini gambarannya. Ini saya menggunakan gambar yang saya gunakan di lab ya, contoh saja. Jadi pertama kalau dalam proses produksi enzim kita preparasi media dulu, kita timbang, kemudian kita autoclave, setelah sterilisasi baru kita inokulasikan pre-culture atau seed culture. Nah, baru proses fermentasi utamanya di fermentor. Nah, tadi terus recovery-nya dengan menggunakan centrifuge. Nah, kita peroleh ini produk enzimnya. Nah, ini yang pertama tadi ya, fermentasi proses tadi. Proses fermentasi ini tadi udah dijelaskan ya, ada seed fermentation-nya, pre-culture-nya, dan main fermentasinya. Jadi proses pre-culture ini memudahkan bakteri untuk beradaptasi dengan medianya kayak gitu. Jadi nanti siap untuk lanjut ke main fermentation. Biasanya untuk mendapatkan enzim yang siap untuk diaplikasikan itu perlu optimasi di proses fermentasinya di fermentor ini, suhunya, pH, agitasi, dan lain-lainnya itu perlu dioptimasi. Kemudian recovery enzim biasanya kita dengan menggunakan centrifuge untuk recovery produk enzimnya. Nah, kemudian purification. Nah, untuk beberapa enzim memang dipakai misalnya dalam kondisi murni. Jadi setelah kita mendapatkan crude supernatant ini perlu dilakukan purifikasi, dan sekarang sudah tersedia banyak sekali kolom kromatografi, beberapa matrik yang bisa kita manfaatkan baik itu untuk rekombinan maupun yang wild type. Kemudian kalau di lab kami ada tersedia akta, ini salah satu alat yang bisa dimanfaatkan untuk kita melakukan proses purifikasi dari enzim. Kemudian terakhir baru formulasi enzim untuk sesuai dengan target dari aplikasi. Nah, ini produk formulasi ini enzim ada yang dalam bentuk bisa cair ataupun dengan dalam bentuk powder. Di LIPI sendiri kita tersedia alat untuk membuat enzim itu menjadi powder. Kita punya spray dry, freeze dry, dan di lab kami sudah pernah mengembangkan enzim powder ini, dan memang aktivitasnya juga nggak menurun gitu, masih tetap bagus gitu. Nah, mungkin untuk khusus pangan, mungkin dalam mendesain produk ini ada beberapa isu, misalnya terkait alergi, terus antimikrobial, dan lain-lain. Nah, ini salah satu contoh aja, saya ambil dari websitenya Novozymes. Ini salah satu contoh enzim protease yang cair, dan ini diaplikasikan untuk pembuatan keju. Jadi kalau pengen kita mengetahui beberapa produk enzim komersial banyak di beberapa website, banyak perusahaan yang produk, kita bisa mengetahui detail nama enzimnya apa, komposisinya apa, kemudian konsentrasinya berapa, dia kelebihannya apa, di cara penyimpanan, cara pemakaian, dan lain, ada biasanya detail di description dari produknya. Nah, kemudian lanjut saya akan bercerita terkait riset enzim di lab kami, terutama yang untuk pangan. Jadi kebetulan kan pusat penelitian biorefinery ini kita selama ini memang udah mungkin 15 tahun terakhir kita melakukan pencarian, screening, discovery dari enzim-enzim yang indigenous Indonesia, baik dari actino, fungi, kemudian bakteri laut, dan juga dari endofitik. Nah, kita punya beberapa banyak koleksi kandidat enzim yang memang sudah high activity dan bisa diaplikasikan di beberapa hal. Salah satunya yang kita kembangkan untuk pangan itu ada untuk produksi oligosakarida. Jadi di lab kami sudah ada beberapa enzim misalnya silanase, pektinis, mananis, amilis, selulase, lipase yang kita sudah punya beberapa strain yang memang sudah kita screening dari beberapa koleksi NACC Indonesia Culture Collection atau Bioteknologi Culture Collection. Kemudian selain itu juga kita lagi ngembangin pektinis untuk di fermentasi kakao. Nah, di amilis sendiri kita punya beberapa kandidat enzim yang isolat yang dipakai untuk produksi malto-oligosakarida. Kita sudah produksi dari beberapa stat yang ada di lokal yang kita punya, baik dari ubi kayu, kentang hitam, taka gitu. Nah, kita sudah, dan papernya sudah publish untuk terkait ini, dan enzim ini dari marine bacteria dan memang potensial menghasilkan beberapa oligosakarida yang nanti bisa dikembangkan lanjut untuk ke prebiotik. Nah, kemudian untuk pektinase kita juga sudah screening dari ratusan actinomycetes dan kita mendapatkan satu kandidat yang dia high activity, dan kita lagi ngembangin untuk ke arah molekuler cloningnya dan untuk scale up fermentornya dengan menggunakan beberapa sumber karbon dari biomasa di Indonesia. Nah, kita di Indonesia itu kaya banget biomasa yang mengandung pektin, di mana ini sebagai sumber utama untuk produksi pektinase dengan biaya murah. Jadi kita punya kulit apel di Malang itu di industri pengolahan apel, kemudian ada juga kulit jeruk, kemudian di tadi sugarcane bagas, di bagas tebu, kemudian juga ada kulit kakao sendiri yang bisa kita manfaatkan untuk produksi. Jadi sekarang tahun ini saya ada riset untuk produksi pekti oligosakarida dari beberapa biomasa ini, dan juga untuk proses fermentasi kakao. Nah, ini salah satu aplikasi dari pektinase kita untuk induksi fermentasi kakao. Nah, kemudian selain pektinis, kita juga mengembangkan enzim mananase dan silanase untuk produksi oligosakarida juga dari beberapa biomasa yang mengandung silan maupun manan. Nah, ini risetnya sudah selesai dan papernya juga sudah publish. Jadi kita sudah siap, ada prototipe enzimnya yang siap untuk diaplikasikan untuk ke area yang lain. Nah, jadi dari mungkin sekitar 500 sampai 700 isolat dari actinomycetes kita sudah mendapatkan satu kandidat mananase dan silanase yang high activity. Nah, kemudian kita sudah cloning dan ekspresi, kemudian udah diaplikasikan untuk salah satunya produksi mono-oligosakarida dan silo-oligosakarida. Jadi dari dua isolat ini isolat.
Mohon maaf sebelumnya Bu Nani, waktu tinggal 5 menit lagi ya.
Baik, baik Mbak Vina, terima kasih sudah diingatkan. Jadi dari dua isolat ini yang sama nama tapi beda isolat, dia sudah direkombinan, kemudian kita manfaatkan dengan menggunakan beberapa biomasa di Indonesia yang mengandung manan maupun silan. Nah, ini contoh produk oligosakaridanya dari mananase. Dia mampu mendegradasi baik manan yang komersial maupun yang ada di Indonesia. Kemudian untuk yang silo oligosakarida, jadi kita mendapatkan satu kandidat, kemudian kita cloning ada dua gen ini, GS family 10 dan GS family 11. Ini juga kita gunakan untuk produksi silo oligosakarida dari sugarcane bagas. Nah, yang unik dari enzim ini, jadi untuk GS family 10 itu bisa menghasilkan silos dan silo oligosakarida baik dari komersial silan yang ada dari sigma, mau ini dari beechwood, bir sama oat, tapi juga dari bagas komersial, dari bagas juga menghasilkan produk degradasi yang sama. Sedangkan yang GS family 11 ini dia hanya mampu menghasilkan silo oligosakarida saja, tidak bisa menghasilkan silos. Jadi mungkin enzim ini bisa ke depannya, kalau memang hanya untuk produksi oligosakarida saja kita bisa menggunakan yang GS family 11. Sedangkan kalau mau silos juga, jadi kita bisa menggunakan GS family 10. Mungkin itu ya Mbak Vina, terima kasih. Silakan, saya sangat welcome. Ini kontak saya. Kalau misalnya saya menerima kolaborasi baik dari swasta maupun dari universitas, terutama untuk swasta kalau memang mau memanfaatkan enzim-enzim yang kita miliki untuk aplikasi berikutnya. Jadi bisa untuk ke pakan, bisa untuk ke bioetanol atau yang lain-lainnya. Terima kasih. Terima kasih Mbak Vina.
Terima kasih Dr. Nani atas presentasinya yang sangat menarik. Baiklah, kita langsung berpindah ke sesi berikutnya, yaitu sesi tanya-jawab. Mungkin bisa di-share pertanyaannya. Oke, pertanyaan pertama dari Ibu Lusti. Pada bidang peternakan, enzim komersil yang diaplikasikan sebagai aditif 90% berasal dari impor. Padahal eksplorasi enzim banyak dilakukan mulai di kalangan universitas, litbang, dan lain-lain. Bagaimana pendapat Ibu tentang potensi enzim-enzim hasil eksplorasi tersebut dan apa tantangannya dalam scale up layaknya produk komersil? Silahkan Dr. Nani.
Ya, baik. Terima kasih pertanyaannya untuk Ibu Lusti. Jadi memang seperti salah satu contohnya untuk silanase ya. Silanase maupun mananase itu kan salah satunya bisa menghasilkan tadi yang saya ceritakan oligosakarida. Nah, oligosakarida ini salah satu fungsinya dia sebagai bisa di industri peternakan dia bisa digunakan sebagai prebiotik dia untuk mengurangi penggunaan antibiotik. Nah, selama ini memang hampir 90% enzim yang beredar di Indonesia kan masih impor semua dan kita belum punya satupun perusahaan di Indonesia yang bisa produksi sendiri dari misalnya isolat yang kita kembangkan sendiri selama ini kayaknya emang belum ada. Jadi kita mungkin sebagai peneliti, saya pribadi emang sangat tertarik untuk bisa kolaborasi misalnya dengan mitra swasta untuk memanfaatkan isolat yang memang potensial unggul ini biar nanti misalnya kita bisa mengajukan proposal bersama. Banyak sekarang funding-funding dari lembaga pemerintah misalnya dari Ristek, LPDP dan lain-lain yang bisa kita manfaatkan untuk menyusun proposal bersama. Jadi untuk mengaplikasikan dari produk kita ke industri kan untuk bisa mendapatkan bukti hasil riset kita itu bisa diaplikasikan industri kan kita harus uji coba dulu, produk kita itu gimana. Nah, ini kan perlu pendanaan, perlu aplikasi. Nah, kebetulan untuk pakan ini saya kebetulan rencana mau ada bimbingan mahasiswa terkait aplikasi untuk diunggas misalnya. Jadi nanti mungkin dengan kalau udah kita buktikan misalnya oligosakarida yang kita produksi selama ini ternyata dia benar-benar mampu banget efeknya itu signifikan dalam mengurangi penggunaan antibiotik. Jadi ayamnya bisa lebih sehat, bobot badannya lebih tinggi. Nah, tingkat kematiannya misalnya lebih rendah. Nah, itu nanti kita dari situ akan bisa mendapatkan misalnya paten. Nah, dari situ baru kita untuk aplikasi scale up di industri secara komersial bisa kita gandeng kerjasama perusahaan yang memang siap untuk kerjasama dengan misalnya lisensi dari produk kita gitu. Mungkin itu ya Mbak ya. Jawaban dari saya.
Oke, terima kasih atas jawabannya yang sangat menarik ya Bu Nani. Jadi kita bisa lanjut ke pertanyaan selanjutnya mungkin. Ini pertanyaan dari Pak Harto Widodo. Bagaimana menentukan bahwa suatu enzim merupakan agen yang bertanggung jawab terhadap suatu reaksi yang kita amati? Bagaimana mengisolasinya sebelum dapat disequence, ditentukan homologinya, dan direkayasa? Dan selanjutnya dilakukan scale up pada host yang sesuai. Silahkan, Dr. Nani.
Baik, terima kasih untuk pertanyaannya buat Pak Harto Widodo. Jadi, untuk mendapatkan bagaimana menentukan suatu enzim merupakan agen yang berjalan. Oh ya, jadi Pak, biasanya kita dalam mencari suatu enzim itu fungsional atau enggak, kita tentukan dulu misalnya. Contohnya nih, saya misalnya mau produksi silo oligosakarida. Nah, dengan biomasa silana kita membutuhkan berarti kan enzim silanase. Nah, jadi kita melakukan isolasi nih dari mikroba yang ada, misalnya dari yeast atau dari bakteri atau dari fungi atau dari aktino, mencari enzim yang isolat yang menghasilkan silanase ini. Nah, itu dengan menggunakan screening, Pak. Biasanya isolasinya dengan menggunakan screening sekalian. Jadi kita biasanya proses isolasinya itu sudah langsung sesuai dengan target enzim yang kita cari. Misalnya ketika proses isolasi, di dalam media sudah kita tambahkan misalnya silan, kalau misalnya kita untuk memang targetnya menghasilkan silanase. Nah, setelah itu nanti kita screening dari beberapa ratus isolat yang kita dapat, nah kita barulah kita misalnya dapat satu kandidat tuh yang aktivitasnya tinggi. Nah, baru nanti dari situ ini kita cloning gennya, kita desain primernya untuk cloning gen penyanyi dari silanase. Setelah itu barulah apa namanya Pak, diekspresikan di host yang sesuai. Kalau misalnya kita dari aktino, berarti kita harus mencari host misalnya dari Streptomyces lividan yang sesuai dari aktino. Kalau misalnya kita isolasi dari bakteri, biasanya kita bisa menggunakan E. coli. Kemudian kalau dari yeast, misalnya kita bisa atau fungi, kita bisa menggunakan host dari Pichia pastoris. Nah, baru nanti setelah proses ekspresi kita mendapatkan rekombinan enzimnya, untuk scale up-nya kita butuh masih optimasi di proses fermentasinya. Mungkin itu ya Pak jawaban saya. Terima kasih.
Terima kasih Dr. Nani, semoga jelas ya jawabannya. Mungkin kita bisa langsung move on ke pertanyaan selanjutnya, yaitu dari Ibu Siti Eka. Jadi Bu Nani, mohon bertanya untuk produksi enzim pangan, adakah titik kritis kehalalannya? Apakah enzim yang sekarang beredar untuk pangan sudah halal semua? Bagaimana Bu Nani?
Ya, memang kalau mungkin kita di Indonesia kan mayoritas Muslim, jadi pasti ini menjadi satu titik kritis dan ya kewajiban kita memang kalau untuk mendesain produk enzim yang memang diaplikasikan di pangan harus perhatian dari awal proses produksinya. Misalnya kalau yang kita kembangin di lab, dari pemilihan media produksinya memang kita sudah memilih bahan-bahan yang halal. Contohnya memang ada yeast extract itu yang dari babi, ada komponen babinya, jadi kita tidak bisa pakai. Jadi ada kalau kita membeli media-media yang memang halal, udah maksudnya aman dan itu memang halal. Ada dari salah satunya dari high media kalau nggak salah, itu bisa dipakai untuk produksi. Kemudian mungkin kalau di lab ya kita kayak Erlenmeyer apa kemarin kita pisahkan, memang nggak bareng dengan yang dipakai untuk yang lain seperti itu. Kalau yang beredar saya kurang tahu, tapi patokannya mungkin di produknya. Kalau produknya udah ada sertifikat halal MUI dan ada nomornya, yang jelas ada nomornya, nah itu berarti sudah halal. Tapi kalau di produk pangannya yang menggunakan enzim itu tidak ada sertifikat halal, nah itu mungkin itu yang bisa kita salah satu cara ya untuk mengetahui kehalalan dari produk itu. Mungkin itu ya Mbak jawabannya.
Terima kasih Bu Nani atas jawabannya. Semoga membantu. Kemudian selanjutnya untuk pertanyaan selanjutnya ya. Mungkin bisa di-share. Pertanyaannya apakah masih ada lagi? Oke. Saya lihat masih ada ya pertanyaan dari link yang di YouTube Bu Nani. Jadi ini dari, oke ini udah mulai screen sharing. Ini pertanyaan dari Bapak Muhammad Zainal Abidin. Jadi Bu Nani, mesin purifikasi akta yang umumnya digunakan ini apakah bisa untuk size exclusion chromatografi? Terus kemudian berapa kira-kira biayanya apabila Pak Muhammad Zainal Abidin COVID-19 ini ingin menggunakannya?
Oh iya, terima kasih untuk pertanyaannya. Jadi kalau akta ini bisa dipakai sih Pak untuk beberapa, semua kolom bisa dipakai ya. Untuk tadi yang disebutkan yang size inclusion chromatografi ya. Ya, itu bisa dipakai. Nah, terkait jasa analisa memang kita lagi di LIPI lagi menentukan Pak harganya berapa. Mungkin bisa dicek di Elsa ya Pak nanti terkait harga. Misalnya mau pakai per jam berapa. Sekarang semua layanan sains baik itu jasa analisa maupun pemanfaatan alat-alat yang di LIPI bisa melalui Elsa di LIPI, di website LIPI ya Pak, nanti bisa dilihat.
Oke, terima kasih Dokter Nani. Semoga membantu ya jawaban dari Dokter Nani. Kita ke pertanyaan selanjutnya, yaitu dari Bapak Patrisius Atmojo di Atmajaya Jogja. Jadi pertanyaannya adalah, kalau ekoenzim itu enzim atau bukan? Kalau enzim apakah? Kemudian salah satu enzim atau multi enzim? Gitu pertanyaannya.
Ya, terima kasih untuk pertanyaannya Pak. Saya pernah dengar sih produk ekoenzim ini ada beredar di pasar, tapi saya pribadi belum pernah ini sih Pak cek produk ingredientnya apa. Jadi saya tidak bisa menjawab apakah itu enzim atau enggak ya. Jadi kalau nggak salah ini salah satu produk yang dipakai untuk apa namanya kayak kebersihan untuk rumah tangga gitu, aplikasi di rumah tangga kalau nggak salah ya, pernah dengar Pak. Cuman mohon maaf saya tidak berani menjawab karena belum pernah ngecek itu komposisinya apanya, terus itu kalau nggak salah diproduksi oleh suatu perusahaan swasta ya kalau nggak salah. Mungkin kalau pengen tahu bisa dicek di ingredientnya. Biasanya kalau produk enzim komersial yang dijual itu akan jelas Pak, misalnya dia nama enzimnya apa, terus kan enzim itu ada ratusan ya, misalnya silanis aja, silanase itu ada misalnya yang GH family 10, 11, 5, banyak. Jadi maksudnya sampai ke isi numbernya itu biasanya ada Pak detailnya. Terus kalaupun dia mix enzim, kalau mix enzim memang biasanya kalau crude enzim dia nggak menyebutkan kalau itu misalnya spesifik udah tadi ya Pak, udah memang enzim yang siap aplikasi untuk aplikasi tertentu, biasanya dia karena dia kan formula. Nah, formula itu kan dia meracik sendiri. Nah, ini terkait paten gitu biasanya tidak disebutkan komposisinya, hanya misalnya nama komersial enzimnya apa, misalnya untuk aplikasi misalnya untuk apa deterjen gitu. Jadi itu komposisi detailnya biasanya enggak. Tapi kalau dia individual enzim biasanya ada detail Pak, informasi nama enzimnya, kemudian unit activity-nya, aplikasinya untuk apa aja kayak gitu. Mungkin bisa dicek di website-nya ya Pak. Terima kasih.
Oke, terima kasih Dokter Nani atas jawabannya. Semoga membantu ya buat Pak Patrisius. Kemudian sayang sekali atas keterbatasan waktunya sesi Q&A hari ini harus kita akhiri. Sekali lagi terima kasih kepada Dokter Nani atas presentasinya yang sangat menarik. Semoga dapat memberikan tambahan ilmu kepada kita semua. Saya juga tidak lupa mengucapkan terima kasih kepada para partisipan yang sudah hadir di sini dan setia sampai acara berakhir. Oke, saya serahkan kembali kepada Mbak Aulia.
Thank you, Dr. Vina. Also thank you to Ibu Nani Rahmani for these sessions, which is the last sessions we have for this afternoon. Distinguished guests, ladies and gentlemen, as we have announced, we have 4 most active participants for today's webinar. The first one is Bapak Harto Widodo, Balai Besar Penelitian dan Pengembangan Tanaman Obat dan Obat Tradisional Kementerian Kesehatan Republik Indonesia, Ibu Lusti Istiqomah, Balai Penelitian Teknologi Bahan Alam LIPI, kemudian Ibu Nurul Izati dari UTS, dan juga Ibu Fauziatul Fitria Pusat Penelitian Bioteknologi dan Bioindustri Indonesia. Congratulations to all the winners. Distinguished guests, ladies and participants, this afternoon, may I kindly invite Dr. Puspitalis Dianti, Director of Research Center for Biotechnology LIPI, to deliver closing remarks this afternoon. Dr. Puspitalis Dianti, screen is yours.
Thank you very much, Mbak Aulia. May I speak in bahasa Indonesia ya, karena yang tamu-tamunya sudah nggak ada. Terima kasih atas partisipasinya Bapak dan Ibu. Saya mengikuti juga webinarnya sampai akhir. Sangat menarik. Dari yang pertama itu adalah basisnya, kemudian yang kedua dari industri. Ternyata juga industri memberikan contoh itu yang mudah ya. Contohnya adalah deterjen, tapi pastinya di balik itu ada ya produksinya juga kan luar biasa kalau Novozymes. Saya berharap tadi juga sudah disampaikan prospek dari industri enzim itu sangat besar sekali. Kita punya kekayaan alam hayati yang luar biasa yang belum dieksplor. Oleh karena itu saya berharap kita bisa bekerja sama ya. Tadi juga dari universitas sudah banyak sekali melakukan penelitian enzim, tapi memang kita itu masih muter, masih screening, paling produksi flash gitu ya, belum produksi besar itu, terus nantinya berhenti begitu. Memang hanya baru discovery ya, kita belum ke arah production. Mari kita bersama-sama berkolaborasi, kemudian fasilitas LIPI dapat digunakan. Silakan, tadi juga sudah disampaikan kita ada layanan sains. Nanti jika ada pertanyaan, silakan menghubungi kami, akan dibantu. Ya. Oke, terima kasih. Dengan demikian, seri 1 webinar di Enzyme ini dengan resmi saya tutup. Kita tunggu seri selanjutnya dengan pembicara-pembicara yang yang bisa meningkatkan keilmuan kita. Ya. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Saya kembalikan ke Mbak Aulia.
Thank you, Ibu. Ladies and gentlemen, with the closing remarks conveyed by Director to the end of the, we would like to sincerely thank, especially for Center of Excellence Integrated Biorefinery Research Center for Biotechnology LIPI, for organizing first webinar of Enzyme series, current and future trends in enzyme technology and its applications. We would like to greatly thank to all representatives from ministries, R&D institutions, and also universities. Thank you to our speakers and moderator for today's webinar. Thank you to all participants who have participated, either from Zoom or LIPI's official YouTube. See you on the next event. Please stay safe and healthy.
Okay, see you. Thank you very much, Dr. Patra Pason. You still together with us. Yeah.
Thank you very much, Dr. Patra Pason.
Thank you,
We
Dr.
will-
Pason.
Please inform your webinar, next webinar or seminar. We will attend it.
Thank you. Stay
Yeah.
safe.
Okay. Stay safe. Allow me to leave. Yeah.
Silakan Ibu, terima kasih Ibu. Terima kasih Bapak Ibu Pusat Penelitian Bioteknologi dan juga para hadirin yang telah hadir. Mohon izin untuk meninggalkan ruangan. Sampai jumpa di webinar selanjutnya.
Thank you.